Es el sonido después de...
Especially for the clapping, like
Yeah.
you need to add. Good morning, everybody. So our second last session already, how time flies. We have 3 speakers as usual, 15 minutes. I will wave at you with 2 fingers when there's 2 minutes left. Hopefully you'll see me. And then if it starts to go too long, I'll stand up and you need to finish within seconds if that happens. We'll have time for questions at the end, so I'll give the audience an opportunity to ask some questions as well. So if you do have any, you can be ready with those. And yeah, I think that's it. So best of luck to our 3 speakers. Up first, we're going to have Aurora talking about phytoplankton communities. So over to you and good luck.
Thank you so much. May I start?
Yes.
Thank you. Well, good morning, everyone. I'm Aurora Menendez Garcia. She introduced me. And today I'm going to talk to you about experimental evolution in phytoplankton communities, how it reduces coexistence but alters community energy fluxes. When talking about species evolution, predicting how species in isolation evolve is quite easy, but does not reflect what happens in nature, where species are in communities. Community evolution is actually a quite not so common topic to approach, as it's really difficult to predict how they will evolve, as they are really dependent of the number of species in the community, the interactions, and the biotic and abiotic factors that they are subjected to. It's been seen that in communities how species in isolation evolve, for instance, while competing for nutrients. It's different than when they are competing in a community, and even competition with pairwise competition, like basically competing 2 species, and competition in communities, it's different. It's been seen in studies of bacteria. Most of the evolutionary work that is being done nowadays has been done in bacterial, with bacteria as model species, but as they are a species that grow at faster rates. They are evolve at faster rates. They reach really high abundances. Therefore, they are have a higher chance of higher mutations. Therefore, evolution within the community. But when we are looking at eukaryotes, we actually don't know if we can use the results obtained from prokaryotic community research and apply it to eukaryotes, as for instance, bacteria have other means of obtaining nutrients which are not present or not commonly present in eukaryotes. We're talking about competition. Evolution with competition, there are 2 macroevolutionary pathways quite researched and established in the evolutionary research community. One of them being the use of different resources, both species which will evolve through character divergence to use different resources under competition, but when competing for essential resources, this may not happen. Therefore, another pathway that they may follow is actually increasing the efficiency. One of the species will increase the efficiency of resource use. It's been seen in studies of bacteria, plants, phytoplankton, so it's quite an established term. Therefore, the aim in my study is actually to see how communities will compete for essential resources, in my case, phytoplankton communities, under 2 different competition regimes, and how it will affect the evolution and functioning of the phytoplankton community. I use phytoplankton as my model species because they are single-cellular eukaryotic organisms that have a high growth rate, and also they reach high abundances. They are a key organism in the marine food webs, and they are also part of cycles in the marine environment, such as, for instance, the carbon pump, the biological cycle. For this, in my research, we've established 3 main hypotheses. We've established that the species should evolve towards a faster growth rate. The coexistence will be favored if a species evolves different traits, and that the co-evolved communities should produce energy and biomass at faster rates. For this, we've created communities of 3 species, having Amphidium carterae, Dunaliella tertiolata, and Thysochrysis lutea. We did a two-phase experimental setup. Phase 1 was the evolutionary phase, which lasted 4.5 months, where we established monocultures of the 3 species. Basically, we introduced monoculture of the species in a dialysis bag. Why a dialysis bag? Because they allow to maintain the monoculture status of the species while being able to exchange nutrients and metabolic products between the bags within the same beaker. The difference between our treatments, which were the intraspecific competition and the interspecific competition, was that in the intraspecific competition, all of the bags within the same beaker were of the same species, while in their specific competition, each bag was a different species. Previously to the phase 2, we actually had a neutral phase, as we decided to call it, which lasted for 5 days, in which we transferred the monocultures from the bags to culture flask. We did this to avoid any plasticity due to the experimental setup in phase 1, which we did not want to affect phase 2 of the experiment. Upon reaching phase 2, we established the communities. We did the communities of 3 species, as I mentioned, and we set them up. Each species had the same biovolume within the community in day 0. Biovolume being a proxy of biomass, commonly used is a commonly used term in phytoplankton research. So for this, we actually, each day of sampling, we sampled for biovolume, cell size, cell shape, growth rate, community evenness, photosynthesis, respiration. These last 2, we used oxygen consumption and production as a proxy to calculate them. From here onwards, I will refer as new community and co-evolved community. New community being the ones that have been, the community has been formed from species that have been in interspecific competition in phase 1, and the co-evolved community from those ones that have been in interspecific competitions. All the data obtained from this was fitted for 4 different growth models, and we chose to represent and do the statistics in the best fitting model according to the AIC. So now for the results. When looking at the species separately, we saw that all of the species grew over the 20 days of phase 2 as the days went by, and we saw that Amphidium was the predominant competitor as it reached the highest biovolume out of the 3 of them. Only 2 of the species reached carrying capacity, meaning they reached the exponential phase, which were Dunaliella and Thysochrysis, but they reached it asynchronously. While Thysochrysis was the first one to reach it by day 8, was not until day 14 that Dunaliella reached carrying capacity. Now we wanted to look, okay, does the growth rate of each of the species between both treatments is the same, and does the species reach for both treatments the same maximum biovolume during the experiment? For this, we saw that actually Dunaliella and Amphidium behave in a similar pattern. They did not show any statistical difference between the maximum rate of increase, which is the growth rate, or the maximum biovolume. But when we looked at Thysochrysis, we then saw that while the growth rate of Thysochrysis remained the same between both treatments, we actually see that the co-evolved community in Thysochrysis had a lower maximum biovolume, statistically significant. Once seeing this, we wanted to see, is the evenness of the community remain the same? How does the treatments behave? We use the Pilow's evenness index, which is an index that represents the diversity of the species richness of the community. It's an evenness that goes from 0 to 1, 1 being the abundance of each of the species in the community is the same. That's why on day 0, where we establish communities at the same biovolume, we have 1. And any dominance of a species, decrease on any species abundance will decrease the evenness. We saw that the co-evolved community decreased at a faster rate than the new community. Having seen this, we said, okay, does the species represent what the community, the trajectories of the community? Is it the same? Does it differ? And when we represented the total biomass of the community, which is basically the sum of all of the biomasses of the 3 species within the community at the same time, we saw that both treatments actually overlap each other more or less in the growth curve, which means that they behave in the same way. We saw that neither the growth rate nor the biomass actually differ between treatments. So having seen this, we said, okay, do they produce more or do you produce less? So first of all, we saw that when we represented the energy production of the community, they behave quite different between the exponential phase, which is the growth phase of the phytoplankton, and the exponential phase, which is when they stop growing, basically. So we decided to split the datasets and analyze them at in different times. In the exponential phase, we analyzed for photosynthesis and respiration, which is basically respiration. And we saw that with an increase in biovolume, the communities increase in energy production. Having the co-evolved community being the most energetic, they produce the most energy, and it was statistically significant only for photosynthesis. While for respiration, they still have the same trend as photosynthesis, but it was not statistically different. They behaved in quite a similar pattern. When we observed the stationary phase, this trend was broken. We did not see any pattern in relation to the biovolume or the photosynthesis and respiration of the communities. Finally, we wanted to see if the energy production of the communities by day actually behave in a similar pattern. We saw, again, similar to what we saw during the exponential phase. They grow over time. They have a positive relation of biovolume with the energy production by day. And the co-evolved communities actually behaved in a similar pattern to photosynthesis. This may be due to the fact that photosynthesis plays a vital role in the calculation of the net energy production. And we saw that the co-evolved community had a statistically higher energy production in comparison to the new community. So I want to take you back to my initial hypothesis. The first hypothesis I presented to you was that the species should evolve towards faster growth rates. We saw that the growth trajectories, as I previously mentioned, of each species were similar. In our research, we expected to have, as we are looking at competition for essential resources, we expected the evolutionary pathway of our phytoplankton would be to increase the efficiency when using nutrients, but we did not see this. Had we seen this, we would have seen a difference between the co-evolved and the new community in the growth rate, having the co-evolved community a higher growth rate as they've been evolving the longest. Therefore, we established that maybe the alternative hypothesis, which was that the co-evolved community, that the species will evolve towards different resource use. Had we seen this, actually, then we would have met our second hypothesis, which was the coexistence will be favored if a species evolve different traits. Nevertheless, we did not see this. What we saw was that the evolution within the specific competitions, therefore the co-evolved competition, reduced the coexistence. This is actually in line with some research that we've seen in bacterial work, where the co-evolved communities actually had a poor reaction to being co-evolving the longer with other bacteria in the community. But when we see these types of work, we have to be really cautious when trying to compare both of them, as prokaryotes have other means of obtaining nutrients, such as, for instance, cross-feeding, where they use waste products as a secondary source of nutrients. Therefore, competition may be hindered, which was actually found in several studies of evolution in bacteria. And finally, we hypothesized that the co-evolved communities would produce more energy as they had been co-evolving the longest, and the biomass will increase at faster rates. Our community biomass production was similar between both fluxes, but the co-evolved community showed a higher energy production during the exponential phase, which, though they have the similar growth rate, actually may tell that coevolution in these types of communities may actually help productivity in the long run. So finally, for you guys, some take-home messages, because I'm talking a lot. So first of all, I want you to get that while we did see some difference in coexistence due to the evolution, history, the traits underlying this effect are not clear. We have to bear in mind that I looked only at biovolume and metabolic rates. So, had I only looked at biovolume, I would have not seen any difference in evolution, and I might have said, okay, my species have not been evolving. Therefore, looking at other parameters is actually really important in this type of studies to discern what happens while a species evolves. Despite the difference in coexistence, the community functioning was more stable. Therefore, looking at species by itself sometimes may not be the best way to reproduce what happens in nature, and more studies in communities should be done. Our results did highlight some classic character divergence pathways in evolution, particularly when species compete in essential resources, and actually coevolution can increase competition and not decrease it. And community function can be more stable if species responses even over evolutionary timescales. Finally, I would like to say thank you to my thesis supervisor, Julia Gadini, for her support during these 6 months, and actually my lab, Dr. Charlotte Tribiden, Daniel Almeida, and Ricardo Stevens for all of their support.
Thanks, Aurora. Well done. Nice presentation. So, if any audience members have a question to get started, you're welcome. Otherwise, we move straight to Jordan. So, yeah, maybe if Lars or Heidi want to start in.
Thank you very much. It's a very nice study. It's the first time I see a study like this. Also, I know that other groups in Europe are working on the same subject, but that one is really nice. Even if the results are not so clear, that's life, huh? It's a science. And I think it's a very important topic. That maybe you do not argue, you had some context and explain why it's important, but I would say that there is another reason why it's very important. And that's about the global modeling of the change in the ecosystem, even in IPCC model. So why is it important?
Why is it important?
Yeah, to know if evolution might change the traits of.
Well, most of the studies, as I've mentioned, were done in bacteria. So different types of species used for models, and empirical work is actually really important for modeling. We do not know how the species will actually progress with different abiotic or biotic factors. So modeling actually is what many of the studies do also apply. So they do the empirical work and they model it. This will have a more clear representation for instance, policymakers. IPCC, as you mentioned. So having empirical work and more data will be able to input it in the models and have higher or more clear results to have a more statistical significance when applying to the real world, let's say.
Yeah, so yes, phytoplankton is in IPCC model, so the model that predicts the change in the ecosystem due to climate change, and also the change in the retroaction from the phytoplankton to the climate, use a fixed value for these traits.
Yeah.
And it's very important to understand how they change, if they change. If they would look at your study today, they would say, oh, we are safe, because it's not changing too much. But I really think that it may change. So I was wondering, you choose 4 months as representative of possible mutations that could lead to a new or change in the traits. Why did you choose 4 months? Is it enough?
At the moment, actually, as we did not see any differences, I believe we should have had more time, because though we did see some difference in metabolic, we did not see significant differences in the, for instance, size or shape of the organisms. We would have if we had time, first of all, because 6 months does not get you much longer, I would have gone maybe for a year. Though maintaining the community was actually pretty hard, especially for the phase 1. They did not like the dialysis bags, so it was quite hard to maintain them alive to be able to see the evolution. Also, the phase 1 of the experiment was done with species that were clonal from the Roscoe culture
Collection.
Thank you, collection. I was not finding the word, thank you. But we maintained them in the lab when they bought them like for a year, so they might have already had some mutations, so that might have hindered already like the evolution part. And also during phase 1, to maintain them alive, we could not deplete them of nutrients, so we kept adding them and reculturing by batch the nutrients. So this may have slowed also the evolution, but there's no physiological, there's no way we could have maintained them without adding nutrients. So we had to maybe sacrifice a bit of time.
Yeah.
And
But from the literature, even on bacteria, how much
actually they evolve quite
would
fast.
yeah.
Bacteria have been seen to evolve within 3 months. Basically, around 100 generations of bacteria have shown evolutionary patterns. And in phytoplankton work, it depends on what you are looking at, obviously. But for what I've seen from generation 70, they've already seen some evolutions. But obviously, if we had more time, more statistically significant results have been found from generation 150
So
on.
how many generations? do you
Actually, we did manage some, more or less. It's more or less at the generation per day. Wait, there. So our phase 1 method. Our phase 1 had 100 generations.
have? 140.
140, sorry.
Okay.
And then during the phase 2, our 20 generations. Had we had more time, maybe also we would have maintained it. Though, as you could see, 2 of the species were outcompeted by day 20, so it started death phase, and they would have died. So it would be it would have evolved towards a single species in isolation, more or less.
Okay. You did all the experimental work?
No, I did neutral phase and phase 2, and I helped the final part of phase 1 because I reached in January, and they already had 3 months. So I did the last month with them, so I learned how to do this part of the experimental work, and I helped in the lab, but if that was not part of my experiment, I wouldn't have had the time to do it if I were alone. So thank you to my lab.
So here you have 7 traits.
Yes.
You did not say how they were measured and.
I did not have time, but I will.
Yeah, I know.
I like that question.
And I think you
Yeah.
made a good choice in your storytelling not to focus too much on the methodology, but still we need to know.
I will tell you now. As you see, biovolume was done with the cell density and average cell volume. Biovolume was actually calculated by cell imaging, so we did use Fiji to analyze. We took pictures of the samples. So thanks to Fiji, we could obtain the cell size, the cell shape of the community, the cell density, and the average cell volume. So that was done with that. Then the growth rate was basically with those parameters, we calculated it, as well as the community evenness, which was basically the Shannon index divided by the species richness in the community, which was 3 in this case. The photosynthesis and respiration, we use actually a quite novel model way of sampling. We use sensor dish reading. So basically, what it basically does is reading through an electrode the concentration of oxygen inside the sample. So they are airtight vials of more or less 5 milliliters. So we put them through 1 hour in the sensor dish reader. So it's a 20-minute light phase where they are under the light with the same conditions, so 60 micromoles per second, which was the same light they were under in the culturing. And we put it there for 20 minutes, then we turn off the light, and we put them through dark phase through around 40. Variations during the experiment, actually this measures the oxygen concentration inside the vial.
Yeah.
Then we do some calculations to obtain the net energy production. They vary, so they normally start at around 100, which is basically when you close the vial. and during photosynthesis they may reach up to 230 to 40. When you turn off the light, they may decrease until 70, 50 we've seen.
The models, among all these traits, they do not use all of them. They use the maximum growth rate and the half saturation constant of nutrient assimilation.
Yep.
Something that you did not measure.
No, for my growth models, what we use is the growth models for biovolume. So
because for them it would be very useful to translate your results into their own type of models.
Yeah.
I was wondering how you could measure, can you obtain the half saturation constant of the maximum growth rate from your data?
I will get so, but right now I'm a bit... for the models, we use 4 different models. They were logistical models, so we use the 3-parameter sinusoidal logistical model, the 4-parameter, and 2 types of Gompers model.
2 types of?
Gompers model. So we use the commonly known Gompers model, and then we use a modified Gompers model with mortality.
Do you have the equation of the model?
Not here right now, but in my thesis I have it. I'm so sorry for that. Yeah. Basically, I can tell you. Okay, so Thysocrisis follow more. All of the samples of Thysocrisis follow the modified Gompers model with mortality, while some of the replicates between Amphibinium and Dunaliella vary in the different models. So each sample, each replicate, follow a different model. So that's then we average, and then we did the interval confidence intervals to show and represent in the graph.
At some point you show a plot with a variable which is k.
Yeah.
What was k? Because I did not really get it.
Maximum variable.
Which is the maximum variable reach during the experiment?
Yes.
Okay. Okay, I know I'm confused because in the global modeling, k is a half saturation constant.
Well, while I was seeing the different models, different papers named them differently. Okay. So you had to see which model you were applying and name the variables after that. But then they use k in different ways, so we just follow. There were other studies in phytoplankton using those type of models, so we wanted to keep it consistent for research purposes after to be able to compare them. Yeah. So that's why we use this.
Okay.
Sorry.
Okay, thank you.
Thank you. Any other questions?
I just have a quick general question. So you did not obtain the results that you expected.
Yeah.
Do you think there's anything you should have changed in your setup, in your methodology? If given the opportunity to do this again, what would you change or do differently?
Maybe add more parameters to look at. See, also I did not do any genetic work. We do not know if there were actually any mutations inside the phytoplankton. Phase 1 of the experiment actually did have, well, it's in the process of obtaining some results for basically the genetic transcriptometry, and also they did DAPI staining to see if actually the nucleus was modified, if they grew or not during evolution, but we have not obtained the results of that yet, so I cannot say for sure. But for sure, having genetic information about it, and also if we would have been able to, maybe it's not the same looking at the community and the species as I told you, and what I have is only morphology and physiology of the species. We would love to be able to split the species by the different, split the community by the different species and run again the SDRs, like the metabolism, to see the actual metabolism of each species. Though for what we've seen on trial and seen in trials, they are under really high stress and they might hinder the information we get after. So it's still, this is a baseline work for this type of experiment, so it's still in research.
It's very important anyways.
Yeah.
Well done, thank you.
Thank you.
I have more questions.
1 minute, 1 minute.
No, but you may have.
No, I don't know.
So why did you choose, or were those 3 species chosen? What did we know about their physiology and their traits before triggers the choice you make.
The first, I will put the images of the species. The first thing is that for our work, as I use imaging to obtain the biovolume and everything, we had to have different sizes of species. So all of these species, as you can see in the scale, actually are quite different size. Amphibian being the biggest, then Dunalela, then Thysochrysis, which was the smallest. This was around 150 microns, up to Thysochrysis, which was around 30 to 40 microns. This was really important so that we could do a really fast analysis of the images. After that, if they are species that are quite common across the globe, so if anyone would like to reproduce this type of experiment with the same species, maybe not the same strain, they would be able to. They are quite common and they are singular cellular and not chain-forming organisms, which was also really important for the image analysis.
Okay. But do they live together in nature?
You can find them together, though maybe not in 3 species communities.
Yeah. Okay.
Thank you. Well done.
That's just minimize.
Okay, so now we move to New Zealand with Michelle. So over to you and good luck with your presentation.
Good morning. Today I will be presenting my master's dissertation on the use of artificial intelligence to assist marine spatial planning. This is the development of a proof of concept study for New Zealand. I'm Michelle Guevara. My primary supervisor is Ashley Roden from Victoria Wellington University in New Zealand, and my secondary supervisor is Danielle Silvestre from Fairwood University, Switzerland. Okay, so as we are all aware, the ocean is facing growing pressures due to anthropogenic disturbances, such as fishing, shipping, and aquaculture. This has led to a loss of natural living and non-living resources in the ocean. Now, as nations aspire to achieve a sustainable blue economy, they need to integrate ocean stewardship, protection, and restoration. And this should be done hopefully through science and technology, monitoring, or what I'll be focusing on today, marine spatial planning. So what exactly is marine spatial planning? Well, marine spatial planning is a process that entails examining and planning where and when human activity should take place in marine areas. This to achieve ecological, economic, and social objectives through a political process. Now, with growing conservation targets in the marine sector, marine spatial planning is gaining interest worldwide. To address and implement marine spatial planning, decision support tools have come into play. One of the mostly used tools for this is Markson. It is a systematic conservation tool, but it optimizes one-time policies. It lacks temporal changes, and it assumes a single gathering of biodiversity and opportunity cost data. An example of the use of Markson is the rezoning of the Great Barrier Reef Marine Park, as you can see on this map. Okay, so to address these gaps that I previously mentioned, Danielle Silvestre and others developed CAPTAIN. CAPTAIN stands for Conservation Area Prioritization through Artificial Intelligence. So CAPTAIN uses reinforcement learning using spatially explicit biodiversity data. It models change in biodiversity over time, so it includes the temporal factor that is missing in Markson and other decision support tools. It also considers anthropogenic pressures in climate change, which makes it highly suitable for marine spatial planning. And it is dynamic and interactive, which means that it adapts to the user needs and the changing environment. So this is why I decided to use CAPTAIN for this project. So the goals of this project were to develop the proof of concept using data and information from the New Zealand region as a case study, to demonstrate how AI can assist in creating a marine spatial plan, to evaluate the effectiveness of the current protected areas in New Zealand, and to identify the priority areas to protect for threatened and protected species to achieve New Zealand's biodiversity strategy targets. Now, to achieve these goals, the chosen study area is New Zealand. So it extends over 5 million kilometers square of the South Pacific Ocean. It includes the New Zealand Territorial Sea, right here, the exclusive economic zone, which is in shaded grey, and the extended continental shelf, right here on the outside in solid grey. Now, the increasing loss in biodiversity has led New Zealand to develop the Te Mana Oceano Biodiversity Strategy Framework 2020. One of the main goals for this is by 2050 to have all indigenous species and ecosystems be safeguarded, and by 2025 develop new tools or collaboratively work with other tools which perfectly aligns with the development of Captain. Furthermore, New Zealand is part of the Montreal Global Biodiversity Framework, whose targets include that by 2030 to restore 30% of all degraded terrestrial and marine ecosystems. Now, these goals that I mentioned are going to be the basis of the Captain analysis. To achieve these goals, I needed a lot of data. So the data layers that were collected were 1 kilometer grid for species distribution models, or SDMs as I will call them now. So these SDMs represent highly suitable areas in yellow and unsuitable areas in dark blue, as well as a 1 kilometer grid for petroleum and mining. Here in pink you can see the active petroleum sites, and in green the active mining sites. This acts as an opportunity cost layer within Captain. A 10 kilometer grid for fisheries data, which I cannot show because it is confidential, but it represents an opportunity cost layer for turtle catch and a disturbance layer for areas swept by bottom trough. Additionally, we included a 1 kilometer grid for marine protected areas, which are represented in a shaded blue right here, and a 1 kilometer grids for dynamic environmental layers of temperature in present and future conditions, and for dissolved oxygen in present and future conditions. Furthermore, lastly, we included a 250 meter grid for bathymetry data, which represented ocean depth. Now, due to the variance in resolutions among these grids, the captain's resolution was set to 10 kilometer grid using a binary interpolation method. And the depth limit was constrained to 2,500 meters, which was the same as those of the species distribution models. Okay, so the species distribution models were obtained from the Department of Conservation's Marine Portal for a total of 583 taxa, of which 239 were demersal fish, 51 were reef fish, and 207 were septile invertebrates, and lastly 86 were macroalgae. Now, one of the key components of Captain is the temporal factor. So we decided to create feature prediction models of the SDMs, or species distribution models. And so a system was created using spatially explicit grid data layers, which included the present distribution model of each one of the species, a static layer for bathymetry, and dynamic layers for temperature, dissolved oxygen in present and in future conditions. So all this information was added to a fully connected neural network with 2 hidden layers, and then the model, so the principle of the model was that if temperature and dissolved oxygen changes, the distribution range of the species will change accordingly. So the system will then observe the environment and calculate a score. The model selects the highest scores after 100 replicates, and it then determines the future geographic range of each one of the species in one time step. Afterwards, each one of the species future distribution models of the species were then used into the Captain prioritizations. Alright, so Captain also, so to achieve the conservation targets that I mentioned, Captain needed, well, conservation status for each one of the species. So therefore, I used the New Zealand threat classification system and classified each one of the species, as well as the Wildlife Act and from New Zealand legislation to determine the protected species. We were using the New Zealand threat classification system, but Captain works with the IUCN ranking. So we had to align these two and give a score from 1 to 5 to each one of the species. depending on how the priority we wanted to give them. It is also worth mentioning that if a species was considered protected by New Zealand legislation, 1 point was given to the overall score in Captain. Okay, so Captain requires the species has specific traits for each one of the species. This to ensure to identify how a species will evolve through time in the system. And the species-specific traits that were used were sensitivity to disturbance, dispersibility, and reproduction rate. For the subtidal invertebrates, the compilation of traits was done using the New Zealand Trait Database and Good 2023. For the demersal and reef fish, the fish trait database used was FishBase. And for the macroalgae, expert input based on the European Algae Trait Database was used. Okay, so now to the Captain prioritizations. With all this information that was compiled, so the present species distribution models, the bathymetric static layer, and the dynamic environmental layers, right here. We included as well the marine protected areas, which I mentioned, the opportunity cost layers, which are the mining and petroleum sites, the fisheries data, which is an opportunity cost layer, and a disturbance layer, the future SDMs that were obtained, the threatened status and conservation status of each one of the species, and the species-specific traits. All this information helped to create a system that was equivalent to the New Zealand region. And well, with all this information, it tells us where and how the population of the species are going to be developed. So the principle of the system was, if the disturbance is removed, the population has the chance to regrow or recolonize. And so the model determines the species that are present in each one of the cells and their respective IUCN categories and protective status. The features are extracted from the system and they are fed into a neural network, which then calculates a score. The model selects the highest scores after 100 replicates, and then Captain, or the model, selects cells for protection and assigns a reward based on the number of species that are protected, weighted according to the species IUCN rank. The reward is higher depending on how good the action was. So, So based on these actions, some populations may recover and some don't, and all this information then goes back into the system for another 10 time steps. Alright, so this is done. We have preliminary results. The Captain metric prioritizations, through the Captain metric prioritizations, we obtained this priority map, where blue shows areas of high priority, dark blue, sorry, and light green or white shows areas of low priority. So the Captain metric was compared to another metric, and it showed that it provided better coverage. Particularly, 30% more species were protected using the Captain algorithm. And protection was mainly focused on the Chapman Rise, which is this area right here, and is also a key area for fisheries, and as well as deeper areas right here. Okay, but these are preliminary results. So what's still to do? Well, these preliminary results can be promising and have already given an insight to what marine spatial planning could look like for New Zealand, but there's still to do for this project. The model needs to be trained using the species distribution models of the macroalgae with the respective species-specific traits. A comparison between the current New Zealand protected sites and Captain's priority areas needs to be made. Furthermore, a statistical analysis of the future prediction models compared to those developed by Anderson et al. 2022 needs to be done, which I forgot to mention are where I obtained the dynamic environmental layers. And future predictions on temperature and dissolved oxygen made by Anderson et al. From 2000 to 2099 should be included as well. And lastly, an analysis of the priority areas that are defined by Captain within the New Zealand region. Now, for the future of marine spatial planning in New Zealand, what's still to do? Well, data. we need data. So we know that AI can predict trends, but we need data to do this. So the UNESCO, with the marine spatial program that they're running, they recommend adding more data on marine and coastal environment, which is partly included, marine and coastal designated conservation sites, oceanographic characteristics and climate, coastal land use and planning, which is not included in this project, operative maritime activities and planning, which is partly included, socioeconomic information, which is not included, and governance information, which is partly included. So this is a step, we're gaining steps. But even with all this, we need stakeholder engagement, which is one of the most important parts of marine spatial planning, if not the most important, and the most challenging. So far, we've done this through the Department of Conservation, Fisheries New Zealand, and New Zealand scientists from NIWA, but we're missing community engagement with the indigenous community, in this case, the case of New Zealand, the Maori, and of course the wider public in New Zealand. This is very important to them, and as well as the scientific community worldwide, which will be done at the end of the year through the International Marine Conference, where I'll be showing these results. Lastly, it is worth mentioning that future forward that we, through Captain, we can use different policy objectives and monitoring strategies to explore various scenarios, which leads to evidence-based estimates. But to further improve this AI and develop a comprehensive plan, reliable and comparable data are needed, along with cooperation with neighboring countries and stakeholders.
Well done, Michelle, thanks. I don't know how many words per second you're gonna Good job, good job. Any questions from the audience yet? Ian?
I was wondering, when you said that with the Captain model, you said that you were protecting 30% more species.
Yeah.
How did the area of actual protected areas and the one that the Captain was given, how do these two areas compare?
Yeah.
Because if it's twice as big, of course it's gonna protect more species. So I was just wondering if there is some kind of difference in the areas.
Okay, so when I said that with Captain 30% were more protected, I was speaking of a metric that was used, but I didn't have the time to explain, which is the STAR metric, which is used with the IUCN. So these were the sites that were chosen by the STAR metric. And this is what I mean that 30% of species were more protected in compared to this metric, okay? Which is based on the IUCN. But yeah, because it focuses on only taking one species, the threatened species, back to least concern. Meanwhile, Captain focuses on doing that, but protecting the species as well. So yeah.
And how these two areas compare? The actual protected areas and...
So the New Zealand, well, we don't want to compare this STAR metric because this is not the goal of the project. We want to compare Captain with the current protected areas, which are these ones. So this is the current protected areas, and this is the New Zealand protected areas. So one factor that I really find interesting is this area right here. As you can see, it is protected by New Zealand, but Captain shows that it shouldn't be protected. Why? Because there's nothing there. So those species are distributed in this area. So, but it is a key area for fisheries. So fisheries have to protect an area. So we chose the one that they would not really need. So this is what I would, for the future, discussing this is very important. So yeah.
Any other questions? No? So we'll move to the jury. Heidi or Lars?
Yeah, thank you. Yes, it was very quick to say, but really interesting presentation. And yeah, even if it maybe seems complicated, it was easy to follow. So very nice work. I understand that you did not submit your manuscript yet, so you are not ready. So my question is about the errors.
About the?
Errors.
Okay, what are?
So when scientists observe on the field, they make errors.
Yes.
When they develop models, they make errors.
Okay.
when they decide of a specific, let's say protection method, they make errors.
Yeah.
How do you combine all these errors to calculate a kind of range of possible maps from your study?
Okay, so if I understand your question is, how do I calculate the errors that
Yeah.
were
Because the errors propagate from the observation to the final map that you
yeah,
observe.
that's very, very hard. But we should start from the species distribution models, which weren't even created by us. But they try to gather as much data as they can, and well, it's better than nothing, to be honest. So
But do they not produce error map? Because usually when you make a prediction map
yeah,
with those models, you can produce an error map.
yeah. Well, we haven't done that yet, but the process isn't
So
done.
I would suggest that something you could do is a kind of what we call a sensitivity analysis.
Yeah.
In that once you have a kind of a standard pass from the data to the end product, you try to vary by 10% maybe some of the parameters of the SDM model, for example. There you could see by how much this map would change.
Yeah, definitely. But for example, this map, it doesn't include the data on the macroalgae yet, because I obtained that today, this morning.
Yeah, probably.
So, yeah, I mean, this is very important to take into account, and of course, we'll discuss this. But yeah.
Yeah, because I mean, if you have to take an informed-based decision for managing this area, you need to know what are the errors and what.
Yeah, definitely. Yeah.
And so I think it's very important that you should look at it.
Okay.
Then, if I understood well, Captain is only the last layer,
No.
the one that combines all the previous steps. Because in the previous steps, you calculate or you obtain through observation map of the unwanted variables. Then you obtain map of the species distribution using the SDM model. Then you have maps of the already existing protected areas. So you have different layers of information. But Captain is only taking them together in a model. So it's the last layer. It's the last calculation you do.
So no, what Captain does is it basically creates a system where all these layers are one on top of each other, so that they align. So each cell is aligned.
Yeah.
So if the temperature in this cell is 23 degrees, and then on top we have the dissolved oxygen for that specific one, and on top so on, and then it all calculates that.
But you could use for example, Marxan is doing the same.
Yeah, but as I mentioned,
And
Captain.
what is the difference between Marxan and Captain?
So this is what I mentioned in the beginning. So Captain only uses one-time policies. So Marxan only uses one-time policies. So in Marxan, you would only protect 30% of the species. That's it. Meanwhile, with Captain, you can protect 30%, but you can also change the protected status of the species. So taking it from threatened to least concern. This does not happen with Captain.
Yeah, but
with Marxan.
What kind of model is Marxan?
So Marxan is an annealing model,
What?
an annealing algorithm.
What does it mean?
Wait, let me remember. So this means that it ranks the data and then finds an optimum. Meanwhile, Captain is a reinforcement algorithm, which means that it takes one input, produces an output, uses this output to then make it an input to produce another output. So it's learning. Meanwhile, annealing is just finding the best optimum among them.
Okay, but with what kind of relationship? Linear relationship, non-linear relationship?
Which one, Captain or?
No, Marxan.
Marxan is non-linear.
The non-linear.
Yeah.
Okay. I think might be a very useful new way of doing everything we want, but it's also very costly in energy.
Yeah.
I was wondering what is the carbon imprint of Captain versus the carbon imprint of Marxon?
Oof, that I cannot tell you right now.
Because I think it's very important.
Yeah, of
Everybody
course.
is thinking that IA will solve everything, but in
Yeah.
my own field of research, maybe Captain, like the IA methods for, let's say, image pattern recognition on image, only brings 5% more of accuracy,
Mhm.
and but the cost of use of energy is maybe 10 times more. And we are at a time that Microsoft is saying that from 2019 to 2024, they have increased their carbon imprint by 50%,
Mhm.
only because of IA.
Mhm.
I think really when thinking of IA, you have to think of also the carbon imprint and to see really if it's useful compared to already what we have. So I don't know if it's planned in your study, but how are you going to compare Marxon and Captain?
So in terms of carbon footprint,
No, in terms of more
that's already been
added
done?
information that is provided by Captain.
so keep or discard?
Yeah, I think you discard. It means you draw.
So that's already been done actually. I have it, where do I have it? Right here. So in a study, Daniel Sylvester, who is the developer of Captain, he showed how Captain was better than Marxon basically. So when they used simulations, it showed for a data set in Madagascar, it showed that Captain had a greater performance of 77.2%.
But what's performance?
So like the conservation areas that were selected.
But
So the performance
which
has been,
criteriums tell you that it's a better case than another one?
yeah, that's what I'm saying. So the conservation sites that were selected by Captain were in accordance with the information that was provided. So it chose better areas because it wasn't focused on cost, which is what Marxon focuses on. Meanwhile, Captain focused on conserving the species, protecting the species. So therefore, it achieved an 18.5% species reduction loss, and it outperformed Marxon. It selected areas of relative importance to actually protect the species, not make it cheaper.
Did he make a sensitivity analysis?
In this one, no, they didn't.
I think when dealing with such complex process that goes from observation to complex modeling, you must be very careful at each step. I mean, developing the IPCC model took 40 years.
Yeah.
Here we are now only maybe 5 years after we started with IA.
Exactly, and this is a stepping stone, as I said. This is the baseline. Like this is just beginning, and I do understand what you're talking about, the carbon footprint, how important it is. But you also have to take into account how much Captain can benefit in marine spatial planning, as this is high seas. We want to include this as well in the high seas, and it's very difficult to do that.
I'll tell you, in my own field, 5% of increase in performance,
Okay.
which I think is not worth the cost
Yeah.
of it.
Interesting. Yeah.
And the developer didn't want to draw attention to it, I'm sure. Do you have a question then?
Yeah, thanks very much for your presentation. It's very interesting to see the evolution of things. First is a general question, is Captain open source?
Yes.
Okay.
It is open source, but
Okay.
it has been approved for the marine environment. So this is what we're working on.
Okay. Because the question that you just pointed out about like we need to have stuff for open ocean and everything, that would depend so much on data that we don't have. So it can only do as much as we can do as well. So there's an aspect to that. Now, in terms of that, so you said your results are preliminary.
Yeah.
Why is that? What are you missing?
So as I mentioned, we're missing data on the macroalgae species distribution models and the species-specific traits. Right here. So this to train Captain, and then the Captain model needs to be trained for around a week. and then it produces the priority areas, and then yeah, that's it.
Okay, so how long does it take after you've input all the data? Is it like a week for
A
it to
week,
work
yeah.
every time?
Yeah. You could run it for 2 days, because this is what we last did for this presentation. So we ran it for 2 days because I had to send it. But then at the end of the 5th day, it was very similar. The results didn't change that much. So it is good to keep it running just in case. But like 2-3 days is enough.
Okay, so your title is about marine spatial planning, but when we are looking at what we are getting out of Captain is areas that should be protected. So marine spatial planning is also about deciding which areas can be explored and how they can be explored better. So how do we incorporate that? Are there plans to incorporate that into your studies?
Of course, yes. That is, as I mentioned, that is the most important part, like stakeholder engagement. This is the most important part. And with Captain, we have something to show. Okay, these are the priority areas we should be focusing on. Let's discuss. Because we have fisheries. They're not going to always say yes to what you produce. We have the Maori community. We have everybody. So we need a consensus from everybody. So the idea is for this to be a baseline. Send it, show it. Okay, let's discuss. Open up a discussion with this. But this speeds up the process because a marine spatial plan can take up to 10 years to be made. So the idea is to shorten that time because we want to protect the ocean as fast as we can.
Okay. And how do you incorporate this feedback from stakeholders?
Well,
if your algorithm is telling you something and your
Yeah.
stakeholders are telling you differently.
Yeah. Well, discussions. There's a board of discussions, and then we tweak things here. Okay, we don't protect these particular species, but we protect these others, and then we see how the model changes. It's once the model is set up, it's easy to change parameters. So yeah.
Okay, thanks very much.
You're welcome.
Thank you. Okay, so we now have our final speaker and the photographer of IMVRC 2024. So over to Ian for your presentation. Good luck.
Thank you very much. Hello everyone, I'm Ian Omedes, and today I'm going to be talking about tropical tuna distribution under climate change. I've been working with Dr. Marina Sanz Martin, Dr. Francisco Javier Abascal Crespo, and Dr. Miguel Cabanella Reboredo at the Spanish Institute of Oceanography in Mallorca in the Balearic Islands. And as probably most of you know here, the impact of climate change, well, you know very well the impacts of climate change in the world, in the water, in the ocean, and as what I'm going to be focusing today mostly is how the impact of climate change is affecting some species in the oceans. So in the oceans, the oceans are warming up, and a lot of species are moving towards the poles. They have a poleward movement, or they are going deeper looking for colder waters, or waters that correspond to the temperature that they were used to in the past before the warming. And when it comes to, I'm going to be talking as well how this is affecting this fishery called the tropical tuna fishery, which is one of the most important fisheries in the world. And inside this fishery, I'm going to be focusing in these three species: skipjack, which is going to be in blue for the rest of the presentation, yellowfin tuna, which is going to be in yellow, and big-eye tuna, which is going to be in red for the rest of the presentation. So these three species make up to 61% of the whole tuna catches in the whole world, and they are essential for the consumption of protein for a big part of the world population. When it comes to numbers, skipjack is number one. Around 3 million tons were fished in 2022. Yellowfin tuna around 1.6 million tons, and big-eye tuna around 0.36 million tons. So as you can see, tropical skipjack and yellowfin tuna, they are considered tropical species, and even if in some articles big-eye tuna is considered a tropical species, since the temperature range where it likes to live, which is around 3 and 29 degrees, which is, as you can see here, slightly lower than the other two species, we are considering it a subtropical species. So most of these fisheries, so most of these fish are being fished by FAD, which stands for fish aggregating device, which is basically a platform, a buoy, some kind of thing floating, which creates a whole ecosystem under it, which attracts other fish and therefore aggregates a lot of a big biomass under it. And these species of tuna are species that usually get aggregated under these pads. In these last years, around, I think I mentioned already, 60% of the whole tuna fishery is being done with FADs. And this is actually very interesting because FADs were implemented in the end of the 80s and the beginning of the 90s, and they have been evolving very fast, technologically wise. They used to just throw things in the water, things that would float, and then come back and hope to find maybe their own or someone else's FAD and fish under them. But nowadays, these FADs have echo sounders that allow them to make an estimation of how much biomass is under them. They have buoys that allow to go straight to their own FADs, making this a very efficient fishery, and well, it basically allows the fishermen to fish faster and more efficiently. So, since it's one of the most technologically advanced fisheries, I had to make an assumption, and it's that the distribution of these species is related with the catches, and well, since it's, as I mentioned, one of the most technologically advanced fisheries, I know it's an assumption, but I think it's quite a safe assumption to make. So my hypothesis is that these populations are being affected by climate change, but I wanted to know more specifically how. And I basically divided it into two parts, a regional and a local part. And for this study, I know sometimes these two words, regional, local, you can use them the same way, but for this study, local means a small area, around 1 square nautical mile, and regional is the whole part of the ocean. It can be the eastern part of the Atlantic, it can be the western part of the Indian, it's a big area. So my two questions are, what are the effects of climate change on the regional distribution? And what are the effects of temperature on the local distribution? So again, regional is a whole part of the ocean, local is 1 square nautical mile, around 1 nautical square mile. So my methodology for the first part, for the regional part, I'm using an indicator, center of gravity, that basically it gives you the mean geographical location of the population weighted by the density. So if this is, for example, the catches of tuna, in each one of these points have a weight of how much was caught in this area, then the center of all of these would be this red point. And for this, I'm using total catch, so how much was caught in kilograms. And if you're interested, there you have the equation used for the latitude part of this indicator. Then going to the local part, and therefore how much each quadrant, each square nautical mile changes, I'm using temporal trend. The temporal trend, basically what it does is it takes all of the quadrants, but now I'm going to be talking about one of the quadrants, and it compares how it changed throughout the years, and it makes a linear regression, and it gives you a trend, if it's a positive trend, negative trend, and for this, I'm going to be using not only, as you can see here, the sea surface temperature, but I'm going to be using as well the percentage of yearly catches by month. It's very worthy, but basically you take how much, how much is fished in one quadrant in a month compared to the whole year. So the data, it came from two organizations, ICAT and IOTC. So ICAT basically is the organization that takes care of tuna in the Atlantic Ocean, and IOTC takes care of tuna in the Indian Ocean. And between the two data sets, I've been analyzing almost 400,000 entries of data. And for the temperature, I've been using Copernicus Marine Service, and I've been using the mean monthly sea surface temperature. And as you can see here, this is the total catches. Oh, sorry.
Is there something?
There you go. Okay, the total catches of tropical tuna since 1993 until 2022, so 3 decades, and that's the time frame that I've been using for my thesis, which for temperature, for catches in the Atlantic and in the Indian Ocean. It usually goes to 2022, but it's fine. So my results, when it comes to the regional part, so the center of gravity, how much the whole population is moving, I found that here you have, in this graphic you have latitude and the months of the year on an average by decade. And if we compare the first decade to the third decade, we can see that the 3 species, as I mentioned before, yellowfin tuna in yellow, big-eye tuna in red, and skipjack in blue, the 3 species in the months of summer in the southern hemisphere, they have moved south, and in the northern hemisphere, in the summer, they have moved north. And there is a larger seasonal migration range, and I have found a significant change in the variance of this distribution. So these 3 species have been moving further away from the equator. Now, in the Indian Ocean, on the other hand, the exact opposite is happening. There is a significant change in the variance as well, and they're moving closer to the equator. So there is a shorter seasonal migration. And for skipjack, what I found is that there is a significant change in the mean latitude. So the whole species is moving slightly north. Now, this is for regional. Going to local, so the quadrants. These are the maps of temporal trend, and as you can see here, in blue is where the temporal trend of catches of skipjack has been reducing. And in red is where this temporal trend of catches has been going up. Here with temperature in red, where the temperature has been rising, and in blue, where the temperature trend is decreasing. And as we can see here, the parts where there is most blue correspond as well to the parts with the highest red, which is around the equator and around the coast of the southern part of the African continent. And I have found during a generalized linear model a negative correlation between the temporal trend of catches of skipjack and the temporal trend of temperature. So there where the temperature trend has been going up, of temperature, the temporal trend of catches has been decreasing. For yellowfin tuna, I have found the same results. Where the temperature rises, the catches are in decline. And for big-eye tuna, the same. Again, now going to the Indian Ocean, I found the opposite. For skipjack, where the temporal trend of temperature was going up, the temporal trend of catches was going up as well. So, and there is a positive a significant positive correlation between these 2 variables. And for yellowfin tuna and for big-eye tuna, I have found no significant correlation between both variables. So, as we mentioned before, the populations of tropical tuna fisheries are being affected by climate change. In the Atlantic Ocean, in a regional scale, they are moving away in the summer from the equator, and in the Indian Ocean, they are moving closer to the equator. And in the local scale, where the temperatures have been going up, the catches have been going down. In the Atlantic and in the Indian, where the temperatures have been going up, skipjack has been going up as well. So it's a little bit counterintuitive. Like they are having different reactions in the different oceans, and that opens a lot of questions. Then that, as you may know, my thesis is still in process, and these are things that we are looking into right now, and we want to see what the reasons are. But so far, one of the main things that we have found is that these 3 species of tuna are amazing swimmers and they have no problem from going from the surface to 300, 400, 500 meters depth. So if they are doing these changes, it's most likely not because they need to go to a colder place, but perhaps because they could just swim down and that would be it. But because they are looking for places where the other two life traits are for other two life traits such as reproduction and feeding. So they are looking for a window of temperature perhaps where their larvae can reproduce better, or they are just following their prey. And the new, for this next week, what I'm going to be working on is adding new variables such as upwelling areas that can be done with using chlorophyll, for example, to assess how the impact of feeding. Or I'm gonna be using as well analyzing fisheries dependent variables such as fishing moratoriums, for example, or expansions of fisheries, or perhaps piracy. And what I'm very looking forward to do is to compare the movement of these species in the different oceans with the movement of individuals taken from data from satellite tags, and I'm very excited to see and to compare these movements that I found with actual data, actual individual movement, and to see if there is some kind of, if my predictions or my results are similar to what the actual data. So I want to thank my supervisor and everyone who's watching online, all of you for coming, and you, the jury, for being here. And as just a last thing, I think it's very important in the future to keep doing this kind of work, not only in a regional level, but there is enough data and it's open to do the same in a global scale, because this is being one of the most important fisheries in the world. It can have it has a huge social economical impact in the whole world. Thank you very much.
Well done, Ian, nice presentation. So again, open for questions from the audience if you have any.
Just at the end, when you were talking about new variables that you wanted to include, I think you mentioned piracy.
Yeah.
How would that?
For example, in the last decades, in Somalia, the effect of how the Somalian piracy has affected the fishing fleet going away from this area, and it can, if they are moving away from a certain latitude, it can have an effect on the distribution that I'm assuming of these species. Or the same with new expansion of fishery. If Oman, for example, develops a huge fishery and it's around the equator, most of it, my results are going to probably show a range diminution. And so it's going to be closer to the equator.
Yeah, because you would have more FADs.
Exactly.
Yeah.
Or the same with some fisheries that went extinct, or other socio-political aspects of the fisheries.
Okay, thank you.
Interesting, yeah. So I pass to the jury. Heidi or
Hi Ian, thanks very much for your presentation. Well done. I hope it all goes well for your finishing up. Can I just find out, what was your sample size in the end? What were you looking at your data? So how much data were you looking at again?
I was using data coming from
from ICAT and
what's the next one? Yeah, here. So basically these organizations, I've as well looked a little bit into others such as IATC in the American continent coast, and basically they all give the data a little bit. It's not exactly the same. They all use sometimes different variables, different names but basically they give you the data of how much is being caught in each degree by fleet, flag, and by kind. But if they're using free swimming fads or other strategies such as free swimming school. So I was using this number corresponds to each one of these lines is a fishing fleet, latitude and a longitude, and a technique used, and what was caught in average in a month in this.
The number, okay. And when you turn that into locally, how much did you have of data?
I basically converted these Excels into a raster, and I was using 1 degree, so
Okay,
each
okay.
one of these corresponds to 1 cell.
Okay.
So
Okay. Okay, so there's a lot of so every time we talk about climate change, we're always associating sea surface temperature, and then we always say things like that are very general, as in maybe they are chasing prey, maybe it is the temperature to do with something to do with their reproduction or their feeding. But do you know anything about their biology in terms of what could this be in terms of tuna is very well studied. So in terms of them moving around, do we know if there's something to do with their biology that actually prevents them from being in this certain?
So as I mentioned before, the temperature range they have as individuals. I mean, I said that they are not being affected by the change of temperature because they can just swim down, but of course there is a limit to that.
Yeah.
For the range of the larvae, I actually found that for the 3 species, the temperature range for the larvae is quite similar as well. So this is why sometimes it's considered a big-eye tuna a tropical species as well, because the larvae can live in the same conditions as the other 2 species. But apart from that, I'm looking right now into their feeding habits, and I'm gonna be looking into that.
Okay, and are you intending to look at their prey as well?
Yeah, and like their feeding habits and like how their prey are being affected by climate change.
Yeah. Okay, good. And then just one more thing. No, actually 2 more things. Sorry.
Sure.
So with climate change, seasonality becomes very fluid because it's very difficult to tell. We end up having a cold or a less cold, or a hot and a less hot.
Yeah.
And what do you think that means in terms of changes for the species of tuna?
So how does the change in the seasons is going to affect their distribution?
Yeah, the fact that the seasons are fluid now, that that there's not there's no limits really. So do you think they would adapt to that since it becomes a bit more fluid in terms of changes, or?
I mean, since I'm using data from monthly data, I think I'm a little bit more safe because, for example, these trends. First, when I was doing this graphs by year, it looked almost flat around the equator because since they were moving north and south, the average just gave me a point in the middle. But then I saw that there was something weird, and I ended up doing the maps by month. So even if the seasons are changing, since I have the data by month, yeah, they can be changing, but then I'm going to see that summer has started a little bit earlier, 2 months earlier or 1 month later. I think it's not safe, but that's something that I think we can pick up when analyzing the data.
Okay. And then lastly, what do your results mean for the conservation of tuna species?
So, as I mentioned, these species have a great socio-economical impact in the whole world, and this is just a first step for I think it can be a greater project. We've been talking as well with my supervisors, and this could actually have, in a global scale, a very big repercussion if we manage to better understand these species, how they move. Because if coastal areas that depend solely in these species of tuna, suddenly the tuna moves hundreds of kilometers away from this area, it can have a great impact. So if we're able to know because with these models, what I can do now, I could take the temperature in the expected temperature for the next decades and if I manage to improve the model with other variables, I would be able to know how it might affect the catches in certain areas, and I think that can have a very positive impact if we're able to predict these changes.
Do we want to improve catches in tuna?
I mean, these 3 species of tuna, they are completely alright. For example, skipjack. One of the reasons why maybe it is not being affected, it's being affected positively and negatively by temperature. It's called the rat of the sea. I mean, it can live everywhere it wants almost, So right now it's reproducing and it has no, right now it's not neither endangered or protected because they are doing honest quite fine.
Okay, thank you very much.
Thank you. Yes, very nice work. My question is about the data themselves.
Yeah.
So the techniques have evolved, as you said, and how is the evolution in the method, in the catch of the skipjack, of the tuna fish, taking into account in those time series?
So what I'm since I'm using, for example, for the local data, I'm using the percentage of catch of the whole year.
It's relative.
It's relative, and therefore I'm protecting myself using these relative, so these percentages. But so it doesn't matter if in 1 year they fished 100,000 tons and the next year 2 million tons, because since I'm using the percentage of how much was caught in each quadrant, it's relative to that same year. So yeah, over a year there might be a difference in the first part of the year and the second part of the year, but in the long run I think it's
Yes, because on the map of the north of the Atlantic, not that one, but the one that goes from 1993 and the video that you made.
yeah.
Here, those videos are not relative; they are absolute because it's
Yes,
total
yes.
catch in
This was just
kilograms.
to present the data.
Okay, because at the end, what I see, looking at that, there is an increase in the total catch,
Yeah, yeah.
meaning that there wouldn't be an increase in tuna fish, which is against your finding that.
That's why I'm using the percentages to analyze the data. So
Okay. Okay. Then, if you look at this map, it's beautiful. We can see that they are not evenly distributed in the area, but there are places where there are more catches than others. So why?
So that's what I mentioned. For example, there might have been a big upwelling in an area, and that's why I'm already looking at different variables to see which ones can have a bigger impact
So
on
which one would you look at?
chlorophyll to see the effect of upwelling and primary production, which would lead to more prey for these species, or these.
Because for a long time it was mesoscale circulation of eddies associated to upwelling and downwelling were thought to be very good explanatory variables for the catch of the tuna. So how could you include mesoscale circulation which might affect the tuna? So will the change in temperature change the circulation of the water and so change the upwelling places,
So by looking directly at the effects of these upwellings, maybe we can skip the step of having to find a way to plot or to represent this mesoscale circulation. So by looking at other parameters that might be affected by this circulation, maybe we can.
I think you should look at, there must be existing modeling outputs of the change in the circulation in those areas. Maybe they could be used.
I will look back the video and check it.
Because what we see here is that so mostly the Mauritanian and Senegal upwelling is one of the main place for catching, and then all the coasts, and then we have the central upwelling of along the equator. It would be very interesting to know if those are going to change with the climate change. And so not only the absolute temperature which might affect the physiology of the tuna fish, but also the distribution of the prey.
But for this, something that I'm doing as well is I'm giving each cell of is it for the total catches in general, I'm giving each of the catches or of the catch line a temperature with coming from the surface temperature from Copernicus, and I'm going to analyze how not only the temporal trend of temperature, that's the variable that I've been using, so how each quadrant has evolved temperature-wise, but what actual temperature, each temperature, what is the effect of temperature in the catches.
Okay. or if I have time,
One more.
maybe you go.
No.
Question. So you spent quite a lot of time to explain this FAD
Yeah.
technique. What I did not understand is these data are obtained also with a FAD, or it's
Yes. So
all the data here is obtained
No.
by using FAD.
this one is the addition of free swimming school, which is basically when the boats are just sailing, and when they find a tuna group, they just go around it and fish it. But for the rest of my data, I just use fish aggregating device, so FADS. And something that in future projects I think has to be done is to combine both data sets and to see how it affects.
How they speak to each other, because so that is not obtained with the FAD.
This is this is FAD for sure, because I did as well the maps just for FAD and just for free swimming school, but I didn't have the time to do them.
Okay.
Again, QGIS, so it would be
Okay.
more beautiful. And what I saw is that FAD, there was a big agglomeration of FAD here, and just under it a big agglomeration of free swimming school, but they were completely separated.
Or just to ensure that there is no methodological bias which might lead to different origin of the data for you, but also to explain for the audience,
Of course.
to reassure.
Yeah, so as I mentioned, nowadays most of the fishing is done with FADs. So it would be interesting and that's why I've used the data from 1990. One of the reasons why I used data from 1993, because that's when FADs were already we have data from FADs from the end of the 80s. So I removed the first years, which had barely no data, so I could have a little bit more confidence in my results.
And then maybe an odd question.
20 seconds.
20 seconds. So if you remove the tuna fish around the FAD, what becomes the other fishes which are the prey of those tuna? What do we think about the change of the community composition under the FAD when you remove the predator?
So how are you relating FAD with the removing of the tuna, sorry?
Because if you remove the tuna, you remove the predator.
Yeah. Oh, and how? Okay, so yeah, so
it's an odd question.
Yeah. During my second semester in Bergen, there was a teacher that was convinced that by removing the bigger predators and moving down we could catch way more than what we're catching right now. So instead of eating tuna, we perhaps should be eating way smaller fish, and that would be a way to feed the planet without. We would be able to have more food for everyone. But it's a little bit, in my opinion, it was a little bit like, yeah, we should kill the tunas, we should kill the big cetaceans, and just be the ones who eat them.
Keep all the small fish for us.
Yeah, exactly.
Too human-centric for me. Excellent. Well done, Ian.
Thank you very much.
Okay, so we have a coffee break now, and the next session starts at 11. So just to wrap up for all three speakers, a last.
of this year's symposium. So we're going to have 3 speakers, and we're going to start talking about phytoplankton assemblages with Hannah Piñel Rodriguez. So you have 15 minutes. When it's 2 minutes missing, I'm just going to try and signal to you, so kind of have a look on the side here. And if you go over 15 minutes, I'll stand up. I won't fight you or anything, but that means that you should probably finish soon. Okay? Good luck.
Thank you for the presentation. I'm Hannah Pina. I'm going to present my master thesis on characterization of algae phytoplankton assemblages, a comparative approach with light microscopy and imaging flow cytometry, supervised by Mark Fernandez Mendez. Phytoplankton are photosynthetic primary producers which lay at the base of aquatic food webs. Their dynamics are related to annual fluctuations of temperature, available resources, and water column mixing, and thus can be modified by climate. Changes in the Arctic Ocean, for instance, are impacting phytoplankton. Typically, flagellates are present through the year in the Arctic and prevail in offshore waters during mid and late summer. Diatoms, on the contrary, dominate at the beginning of the season when high nutrients are available. To assess changes in phytoplankton community composition, time series with a high temporal resolution are needed. Traditionally, taxonomic identification has been carried out with microscopy. However, this is time-consuming and requires high expertise and taxonomic knowledge. It is observer-dependent and prone to error. With it, the identification of the taxa is impossible at the scale needed to assess the current state of the oceans. Because of this, some automated systems have been developed in the last decade, such as flow cytometry. In this case, particles were in suspension, pass through a laser beam, and they get sorted out based on their fluorescence properties. However, this has low taxonomic resolution because there is no image attached to each of the cells. A new emerging technology could change this, overcoming limitations of both methods. Imaging flow cytometry combines the statistical power and fluorescence sensitivity of flow cytometry with the imaging capacity of microscopy. There are several systems, however, only the AMNIS image stream can take both bright field and fluorescent images simultaneously. The identification to a species level is possible in the case of culture known species, but not yet for natural complex samples. To assess its applicability for this, it is crucial to compare an IFC approach with traditional microscopy. Just because in this thesis we will compare the number of individuals detected with both methods to assess if it will be feasible to directly quantify natural samples with IFC in the future. Some of the hypotheses are that microscopy will be more accurate than IFC in quantifying phytoplankton species from these natural samples, that due to the necessary different volume analyzed, rare species will only be detected with microscopy, while IFC will provide a more accurate and unbiased quantification of the most dominant species, and that in the phytoplankton community will present a high abundance of flagellates during the late summer. The sampling took place in the Central Arctic during the Earthwatch 1 expedition on board the reserve vessel Pelastian during the late summer of 2023. Some samples were taken at the chlorophyll maximum at ice stations, and samples taken from stations 9 to 16 were collected within a transect from the vertical integration of water from 2 to 50 meters. Finally, here in red, one sample was collected in another ice station. The samples taken at the ice stations were collected with Niskin bottles attached as a rosette to the CTD. They were concentrated using a reverse filtration device which allows gently concentrating many liters of water into a small sample. They were also passed through a 100 micrometer mesh to exclude tracers and mid-size restrictions for the IFC. The samples were fixed in formalin and stored for further analysis in the laboratory. In the case of microscopy, 3 milliliters of seed sample was sealed in an open chamber, and they were analyzed with the inverted microscope with phase contrast. The cells were counted with an electronic counter. Heterotrophic dinoflagellates were also very abundant, and thus was also counted as well as other microzooplankton. A
Additionally, samples were acquired with the AMNIS image stream. A very small volume of up to 100 microliters was inserted into an dependent that was put into the machine. And once in the IFC software, these three files were merged together because they go down 3 times per sample. The channels of interest were bright field, fluorescent emission of chlorophyll A, and side scatter, which is a measure of texture. Grading is the selection of successive populations for analysis, and it was carried out in a semi-automatic way. For the comparative analysis, all counts were calculated per liter. The percentage variance between methods per taxa in each station was calculated using the microscopy value as a reference. An image was used for some measurements. In general, the total abundance acquired with IFC was considerably lower than the total abundance obtained by microscopy. However, IFC could acquire some very small cells from 2 to 4 micrometers that were not accessed with the microscope, and also photosynthetic cells from 15 to 30 micrometers that could not be identified. Of the total 121 taxa that were identified with microscopy, only 75 could also be seen with IFC, including size classes and unidentified morphotypes. Now, analysis with the imaging flow cytometry, typically with the AMNIS, typically covers a particle range from 500 nanometers and 100 micrometers. However, cells up to 250 micrometers could be acquired with a good focus. Even if there was an initial limit with the mesh of 100 micrometers, thin long cells could pass through by their longer axis, and the software can automatically merge 2 images taken into one. Identification of differentiation from dead and alive cells with microscopy can sometimes be difficult, but IFC proved to be very efficient in this aspect, differentiating between dead and alive cells based on the presence of chlorophyll. If it were dead, this will be black and we will see no signal. IFC proved to have some difficulties in identifying certain species. In the case of centric diatoms, for example, Asteranophyllus could not be identified when alive because focusing up and down with the microscope is necessary to see these rays, which are normally covered in chlorophyll. In the case of Cathoceros or Phytoceros species, they have long setae, which are needed for identification. So IFC was not really suited for this because of their big size. Here we can see the variance percentage, the mean variance percentage for the main groups for all of the stations. As we can see, IFC underestimated all of the phytoplankton groups in comparison with microscopy, except for Phytocystis. However, there is high variance in between stations within the same taxa, so we need to see this more in detail. Here we can see the variance percentage for diatoms in each of the stations. In the case of centric diatoms, they were underestimated in all of the stations except for the last one. Penny diatoms were underestimated in all of the stations. This high variance found in station 9 is due to Cathoceros species, mostly Cathoceros socialis, which was found in high abundance under the microscope, although IFC revealed that the majority of the cells were dead. The same situation was found for Pseudonychia and for unidentified pennies under 10 micrometers. So this can tell us that IFC seems to be more successful at identifying cells that are really alive. In the case of dinoflagellates, Attecate and Tecate were underestimated in 13 and 14 stations, respectively. They were mostly heterotrophs, or they were manually selected as populations with the IFC software. Even if distinguishing autotrophic and heterotrophic or mesotrophic species can be difficult in microscopy, especially for morphotypes, IFC proved to be really efficient at distinguishing based on the fluorescent properties of the images. Silicoflagellates were very abundant in all of the stations, but mostly dead, like this one. However, as in the rest, only the life forms were taken into account. IFC was very successful at identifying them because of their very distinct shape. Biocystis was the most notable difference between methods. This aptophyte has two major morphologies: solitary cells, or cells embedded in a gelatinous matrix forming large colonies. Only solitary cells in flagellate state were observed. This high variance percentage that we can see in some of the stations can have an explanation. When one taxon was very abundant, it was counted in a small section of the chamber and extrapolated to the whole area of it. In the case of Biocystis, this was done in stations 5, 6, and 12 and 10. In the rest of the stations, the small Biocystis cells were counted one by one, and this led to an underestimate of their abundance by human bias. With IFC, we can see both the bright field and fluorescent images of the cells, which makes their identification easier and more reliable. This leads to believe that IFC might better represent the real abundance of this genus. The under ice station, the sample taken at the under ice station didn't have an initial volume because it was an algal aggregate. So the relative abundance was compared between taxa. Fragilariopsis was the most dominant species, and it had to be counted within a track with the microscope because it was overlapping so much. But we can see that with IFC, the relative abundance was much higher than with the microscope. If we exclude Fragilariopsis, we can see that after other primates, Arteria was quite abundant, a little less with IFC. This species is adapted to low light and colder temperatures or residing under ice. This explains why it was only occasionally detected in the CTD stations. Now about the phytoplankton community composition in the Arctic. The results agree with other past studies that found that the majority of the phytoplankton community in the late summer in Central Arctic and the surroundings was constituted of a relatively small and mostly unidentified flagellates, as we can see here in blue. This was the exception only in the stations closest to the North Pole. This suggests that a late summer bloom of Catocera socialis occurred. The station closest to the shore of Svalbard had the highest abundance of bigger diatoms belonging to the microphytoplankton. All of these can agree with the initial hypothesis that flagellates prevail in offshore water during late summer over diatoms. Also, in this first station, Pseudonychia was the most dominant species. It was sampled at the beginning of August. However, this species is known for all-year blooms with peaks in late August or early September, suggesting that an earlier bloom took place. Now, to conclude, in general, identification with IFC was achieved to the same taxonomical level as microscopy in general. Microscopy was more successful identifying the whole diversity of the sample, but IFC might be better at representing the real abundance of high abundance species as Biocystis. IFC proved to be a very valuable tool for distilling alive from dead cells due to the presence of chlorophyll, as well as inferring feeding habits from the same characteristic. The groups that were not observed with IFC comprise mostly the cells inside the bigger size classes and rare species. And finally, the analysis of the sample from the Central Arctic in the late summer was mostly a flagellate-based community, followed by abundance by diatoms. It will be really interesting to replicate the study with a higher total volume per sample with IFC. High throughput quantification of phytoplankton communities could speed up sampling for detecting changes in the community composition within a broader change of time. And the developing of a trained algorithm to aid the IFC analysis could facilitate the process. The image base gathered in the study can be used to train an automatic training algorithm for phytoplankton recognition. This is all. Thank you so much for your attention.
Very much, Anna, for your presentation. Do we have any questions from the audience? Yes, Ian?
Can this technique be applied to other areas or is it more specific?
Yeah. Actually, the AMIS image stream is a machine that has been used for biomedical purposes or medical purposes because with other channels you can also see a stain, for example, nucleus or whatever, and it's been recently starting to be applied to phytoplankton. There is one researcher in Germany who started with phytoplankton but from not marine environments. And it's like something starting to be talked about. It hasn't been done before in this case, so.
Anybody else? No? Okay, so I'll give our panel the opportunity to ask some questions to Johanna. Who wants to start?
I start? Yeah, sure. Okay. So, congratulations for the work and thank you for the very clear presentation. Okay, it was you did prepare a nice presentation with images and so supporting materials and results. So it's congratulations for that work. I have talked about the utility of these tools, especially when you are doing a taxonomic analysis. No. No, okay. The thing is that, in your opinion, okay, do you think that how we can overcome is the gaps that you found? At the end, you propose just improving the algorithm, training the machine or the system, but you all indicated that there are some particular species that really they need somebody just in front of the microscope to identify. At some point, you mentioned that the main problems or gaps were with the, let's say, bigger size species and those that are more rare. That in some cases are the interesting ones, not those that are so what are the next steps to see that it could be able to replace
Well, I believe that I mentioned this in the thesis, didn't have time here, but maybe mixed analysis will be good because as we saw for phylocystis, it's really difficult in microscopy when you have samples from different stations, but it is very abundant in one, and because of the methodology, you count it in tracks. If not, it wouldn't be possible. And in others, maybe you have 10, let's say, and you might see maybe 8, but you might miss 3 because you have to look at the whole chamber and it's a lot at 4D magnification, 1.6 optical. So in these cases, IFC might be more reliable to say the real abundance of these species, or maybe of even low-size species that are very abundant in general. While with microscopy, you have to look at the rare species for sure. But it is, I think it could be really useful because, for example, in Helgoland they have done series for, I think, more than 20 years, and they do one sample each day. It is impossible to make replicates and everything, so maybe even you cannot look at the whole diversity, it will be very useful to have an analysis of IAC of volumes taken each day because it is so much quicker than microscopy. So it will also be nice to start trying to do this just to see specific things like maybe when an algal bloom is coming. In general, the composition of the community, diatoms or dinoflagellates, or maybe to genus level instead of to species level. So this could have applications in the field.
For example, what is the speed of the analysis? I mean that when you go, I can imagine that maybe one day you can proceed several samples.
The speed, when you have to put this 100 microliters at max, that acquisition takes most an hour. I did that 3 times, so it was 3 hours for one station. and this the analysis takes from 1 to 3 hours, let's say. So it's faster. With microscopy, it takes, for example, like from 1 to 2 full days.
Okay. No more questions. Thank you.
Okay, thank you. Nico?
Yeah, well, congratulations, Hannah, for this presentation. It was really good. It really flowed very well. I think you made an extra effort in explaining the difficult part, like how the cytometry works and all that. So congratulations for that. And about the content itself, I agree with you that to do microscopy with phytoplankton could be very important to see the specific and very detailed taxonomic composition of the samples. Yes, I've been working with scan.
Yes.
I compare that with yourself at the microscope, and it makes a difference. It really makes a difference, especially telling apart the species that are very, very similar. But on the other hand, as Madame pointed out, you go very slow. So yeah, probably what do you think that if you have to make a sampling, how would you combine these two different automatic methodologies versus more traditional methodologies? How would you combine that in a sampling?
I think that if I had to do that, I will do at least 1 sample per station with microscopy, and it takes 1 day, so you have the full detail of the sample. But then don't look into, for example, species under 2 micrometers. Don't look for the nano microphytoplankton. I will do that with IFC as well for Phyllocystis or very small species. And Catoceros species of like Socialis that I put in Mister were very easy to identify also because of their shape. So it is also very useful for that instead of with microscopy having to do tracks, and you always have an error associated that can be quite big. So I will make the bigger species with microscopy and the smaller ones with IFC. And I think after trying that with trial and error and see that indeed IFC is better at identifying Phyllocystis for having a more reliable database.
I was a little bit surprised to see that there were not statistics here, right?
Yeah, it was not possible because for statistics I will have needed replicates. In the case of microscopy, I did just 1 sample per station, and because if not I wouldn't have had time in the 4 months that I was there. And in the case of IFC, I did 3 replicates per station, but then I had to put them together for analysis because sometimes the counts were really low, like maybe in one it was 300 cells in total for the sample that had chlorophyll. So it will be the next step to try to do this with more time, make replicates with microscopy and with IFC, because it will have taken triple the time to do the work.
Yeah. Probably you should have thought this beforehand, so you could say, okay, I'm going to reduce my spatial coverage, for example, so I take less stations, but in these stations I'm going to replicate, because replication is important to assess the results. Because I see in those graphs, it's kind of apparent that there is a difference in the number of species and total abundance. But it's a pity that you don't have replicates to show that statistically, that at the end is very important.
Yeah, it was not possible because we decided to put before the special resolution to have more stations to see in all of the stations that they were, instead of focusing in less stations and doing that in multi-day.
Yeah. Finally, one question about the flagellates. You say that they dominated at the end of, or the middle end of summer. What do you think is that?
Because as I said at the beginning of the pelagical algal bloom, which takes place when the ice basically is when the ice retreats in the summer, the algae grow very quickly because of the stratification and because of the nutrients that they have. So then we have many diatoms, but then as they grow and because of the predation and everything, then flagellates are much more abundant with the less nutrients. So it was very nice to see that these results were the same as many other studies in the area. And they were mostly small flagellates, but also flagellates as, for example, dinoflagellates, many species.
And they are not supposed to do strictly photosynthesis, right?
Yes, mostly they were heterotrophs. So, and that was what I expected. So.
Okay, thank you very much.
Okay, Hannah, I was actually going to ask the same thing as Nico because I didn't see any statistics there.
It was mostly like a methodology development-based work. So to see if the massing could be used for this.
Yeah, but when you were comparing a methodology with the other, you're saying, oh, this, overestimated this species and this underestimated this species. It doesn't actually mean anything until you statistically see if that's significant or not, So definitely, this was one of the points that you needed to have prepared for, because we see a lot of students that have a small sample size, and we are like, that's okay, that's all you got, and we need to be careful looking at the results, but we need to see that you know how to analyze these results, and take the limitations into consideration. So definitely, if you're continuing this kind of study, in your design, you have to prepare for that in advance.
One possibility maybe would have been to do an NMED analysis of the phytoplankton populations in between microscopy and the amnis, but because the species level was very different, as I said, some species could not be identified to the same level. Maybe it would have been very general groups, like also dinoflagellates or centric diatoms, so the NMEDs wouldn't have been really accurate. So I decided not to get into that and focus más in the comparison between methods, which was the main point of the work.
Okay. So for the people that don't work with that, I think you should have specified a little bit, and if you could do now, how the IFC works. So I understood at the very end, and I think when there was a question that it is a library, but you said also something about training it with AI. So could you just elaborate a little bit how that works?
Yes, the training with AI will be a possibility for the future because it is not completely developed yet, so they will have to be trained for phytoplankton if they use it in medical field. But what the amnis or NEIC does, it's basically the same as flow cytometry. The cells go through the laser and get sorted out, but in this time there is also a camera. Sometimes in other model that is even more complete, it is two cameras, and it takes a photo of the cell as it passes, and also different fluorescence. So for example, I work with chlorophyll A fluorescence, but you can also stain the cells, you can stain a part of the cell and see how it looks in the photo. So it has a lot of applications.
But it does work through a library that was fed into it.
No, I did the analysis. So in the IC software, what happens is that you have different features for the cells. So for example, intensity of chlorophyll, the layer axis that you can put in all of the channels that you want, compactness, love count that I use for phytocystis. And what you do is you use these features to create graphs. It's the right way, but to create graphs. And with that, later you can see you can create regions based on that. You can see that, for example, phytocystis is in a region, then that creates a population. Normally, with flow cytometry, for example, you create the region and maybe you have something inside that is not part of it. But because of the image, you can click in one point in the graph and you have the photograph of what you are seeing. So because I was comparing it and I wanted to be accurate, I made sure that everything inside the populations was within the group that I was trying to select.
Okay, thanks very much.
Thank you.
Okay, everybody, so welcome Julia. So now we're going to listen to Julia talking about grey whales. So whenever you're ready.
Thank you.
Good luck.
Welcome everyone, good morning, also online. So this is my presentation titled Navigating Nutrition, Impacts on Reproductive Success in Eastern North Pacific Grey Whales. My name is Julia, and my supervisor was Dr. Joshua Stewart from the Marine Mammal Institute at the Oregon State University in the United States. So grey whales undergo an annual migration of 50 to 20,000 kilometers. This is actually the longest migration of any mammal in the entire world. And they feed in the Bering and Chukchi Seas, which is in Alaska, and then they do a southbound migration to Baja California, where the breeding areas are. And then they travel back up north, which is called the northbound migration. So in the Bering and Chukchi Seas, grey whales occupy the space from May through October. They primarily feed on benthic crustaceans, and they use these to build up large energy stores that are very crucial for their migration, because this is actually the only time when they will be feeding. Then they embark on their southbound migration from October until December. Those are first pregnant females, then breeding males and females, and lastly the juveniles. In Baja, they will give birth to a little baby, and the pregnant females, and they will stay there from December until February. And the non-pregnant females and males, they will breed, otherwise they will calve. And then they embark on their northbound migration from February until April. And here we will first have the males and females without calves leaving the lagoon, and then the cow-calf pairs, which will actually stay pretty close to shore because they're trying to avoid predation of killer whales. And most whales decrease in body weight 11 to 29% during this migration because they don't feed. And this is especially crucial and important to remember for the rest of this presentation for the lactating females, as they have an extra energetic burden of lactation. So the Eastern North Pacific grey whale population has been exposed to some unusual mortality events. One from 1909 to 2000, and one from 2019 to 2024. This most recent one was actually just declared over in March of this year. The unusual mortality events have been linked to starvation and poor body condition of grey whales that are then further linked to prey availability in the Arctic. And previously, it has been hypothesized that it might be linked to carrying capacity, but more recently, they're more leaning towards the prey availability. So we were asking two different questions. The first question is, how do different life stages respond to this fasting period? And our objective here was to assess the nutritive condition among calves, lactating females, and other whales. So this means other whale, like the adult whales. And our hypothesis is that lactating females and other adults decrease in body condition, but calves will increase in body condition as they're feeding on their mother's milk, which actually consists of approximately 50% fat. And our second question is, how does body condition impact reproductive rates of females? So here we're trying to explore the minimum threshold for females to be able to successfully reproduce. So moving on to our methodology. So we have 3 different sampling locations. There's only very limited data available from the Alaska surveys, so most data is from the southbound migration. This data was collected by the National Oceanographic and Atmospheric Administration, also called NOAA, and the Marine Mammal Institute is cooperating with NOAA, so we're able to use this data set. For our calf counts, these are done from shore, so they're just being counted. There's no body condition data, so it's actually a much larger data set for those. And the data from 1987 to 2013 was collected using fixed-wing airplanes, and they primarily measured maximum width of the whale and length, and then later on, from 2015 to 2019, we changed it to using drones, and we started measuring width at 50% length and the total length of the whale. So body condition is then measured by dividing maximum width or width at 50% length by the total length of the whale. And during both surveys, sometimes both of these measurements were taken, which allowed us to create a correlation graph and to transform some of our maximum width values to 50% length, which is a more standardized measurement that can be repeated more easily. So for a body condition between years, we're expecting it to be fluctuating depending on the prey availability in the Arctic for our objective 1. And then further for our objective 1, within each year, we're looking at pregnant females, lactating females, and calves for each year on the southbound migration. So we're still expecting pregnant females as they're just heading north to their breeding areas, but we will have some females that will have already given birth to their calves. So they will have a higher body condition compared to other whales, which is our baseline at 1, because it's a proportional measurement. And then calves will have a lower body condition because they will have just been born. So then for our northbound offset, so calculating in our Bayesian model from our southbound migration to the northbound migration, we're expecting other whales, meaning the adults, to be decreasing as they're fasting, and lactating females to be decreasing the most because of the energetic burden of lactation. And calves, we're not exactly sure because they might be increasing in length rather than width. Then for our calving threshold. So there has been some evidence of body condition influencing calf growth, but our main question is if there's a minimum threshold for females where they're not able to reproduce anymore. So we're looking at cow-calf pairs on the northbound migration, and as this data set is limited, we are only considering years of more than 10 data points for lactating females. And then for each of these years, we're also categorizing them into low, average, and high calving years. So those are the calf counts that are done from shore. Now, where is this? So for the mean population condition, so we have body condition on the y-axis and then our time series. And you might be wondering why there is a major dip here that I didn't mention. And this is actually hypothesized to also be an unusual mortality event, but it was not officially recognized as one. Then from 1999 to 2000, we have one of the mortality events, and then the last one, that is just, that's just getting started. So we are in 2019, so we're not seeing the major dips just yet, so because we don't have any further data here. So then our proportional changes, so we have our baseline here. This is the comparison from the southbound to the northbound migration. And we have found that calves are increasing by around 10% in body condition. Cows, meaning the lactating females, are decreasing by around 20%. And all the other whales are decreasing by around 10%. Now for the calving threshold. Don't be scared, I know this might be a little overwhelming, this graph. But you might be wondering why there is a gap in between here, but that's because we have consecutive years here and consecutive years here, so I didn't really want to connect them. But to explaining the graph, so we have body condition of the lactating females and our years, and then I divided each year into the different calving years that can be seen here. And then to figure out our calving threshold, I explored the mean, minimum, and 2.5 quantile. So up here we have the mean, then the 2.5 quantile, and lastly the minimum. So we settled in the end in the mean of the 2.5 quantile, which is this line, and it is 0.145 for the northbound migration. So if we apply this threshold to our data, we can see that there is a clear trend that most females are above this line, the females that are with calves. And then in our Bayesian model, we're back calculating this threshold with our proportional offset to see what their body condition would have to be in Alaska. So when they're just starting on their migration. And this will allow us to then predict future unusual mortality events. So, my discussion. So what did we find? We found that lactating females have double the proportional decline in body condition compared to other whales. And we found that body condition is a valuable indicator of reproductive output because females with a lower body condition are not able to have a calf. So the stage-specific changes we found is that lactating females have the largest decrease in body condition, which is very similar trends that have been observed in North Atlantic humpback, Sei, blue, and fin whales. And this decrease in body condition can actually lead to the loss of a calf or even prevent reproduction overall. And the energetic cost of lactation is very high for these animals because they're not feeding. Then for calves, they're increasing in body condition because they're able to feed off their mother's milk, but they're very dependent on their mother's size. So if there's a female mother that is very slim in body condition, the calf will likely also be very slim in body condition. However, a rapid increase in body condition is very crucial for calves as they're just embarking on their first migration up north, and it will take another couple of months before they will be able to feed for the first time. And then birth rates have also been linked to crustacean biomass in the Arctic. And lastly, our adult whales decrease around 7 to 11% in body condition, which similar trends have actually been observed also for humpback whales. So then for unusual mortality events, these have likely been have affected rates, reproductive rates and survival of individuals. They're also linked to low prey biomass in the Arctic with high ice cover, so sometimes whales are not able to reach their feeding grounds. And this has led to a decrease in 15 to 25% of the entire population. And also previous studies have confirmed that body condition influences reproductive success. And then for specifically climate change, the prey distribution and abundance of their prey is very crucial for them. And they might be physically excluded from their feeding areas. Might have implications for reproductive success and population decline. Some studies have suggested that climate change might impact their migration timing, as they might also have to travel further north to access high prey quality, and then obviously travel further south to reach their breeding and mating areas. And lastly, as this whole presentation kind of underlines, feeding is very crucial for reproduction. So now, kind of going back to the beginning, our two questions here combined: are there stage-specific differences in body condition, and how do these impact female reproductive success? So our first objective was to assess variations in nutritive conditions among calves, lactating females, and adults. And we found that lactating females have the highest decrease in body condition, followed by adults, and calves increase in body condition. And then for our objective 2 was to explore the minimum threshold for female reproduction. And we proposed a minimum threshold of 0.145 for their southbound migration, which will be, for the northbound migration, which will be higher in the Arctic. All right, that's it. Thank you very much.
For a very nice presentation. Do we have any questions from our audience? Yes?
I'm just curious, can you tell from the plane which ones are males and females if they don't have a calf?
No, you can't. So those would be categorized as other adults, yeah.
Any more questions? No? Okay, so next one to you guys.
Okay. Okay, Julia, congratulations for this beautiful presentation. I really enjoyed the presentation. It was very well done. The transitions, the drawings, the photos, all of that was nicely assembled, so you can actually get a feeling of what you did. So congratulations for that. And about the content itself of your work, I think you made a very good job showing the different statistics and so on. I just have very few questions. Well, one observation or curiosity, just at the beginning of the introduction, you said that these are the mammals showing the longest migration on Earth.
Yeah.
Humpbacks. What about humpbacks?
Yeah,
From the Indian Ocean all the way to the Antarctic and back. That's a long distance. I bet we should measure that, but I bet it's longer than the other. That's a curiosity.
Yeah.
Then there was, at some point, I think there was some confusion about two different terms. You use the term trend, but actually I think you wanted to say pattern, which is not the same because you have not found any trend, right?
Mm-hmm.
It's not that the body condition worsen with time, right? You haven't found that.
No.
Actually, it stays pretty high from the beginning to the end of the time series.
Yeah, it fluctuates.
It fluctuates?
Yeah. But yeah, so like during unusual mortality events, yeah, you will have lower body condition.
But there is no trend. So how is that fitting or coupled with climate change? Do you think that climate change has any effect on the body condition of these animals?
Yeah, I think so, because their prey is actually moving further north, so they're high quality prey. So when individuals feed in their current areas or in their previous areas, they were able to get better quality prey, but now they have to move further up in the north, so there might still be some more ice, and then their migration will take longer.
But that's, if that's true, you would have seen a trend.
I mean,
No?
that's a prediction for the future.
For the future?
Yeah.
Not
Yeah.
for now?
No.
So for now you cannot say that?
No, I can't.
Okay. Just to be clear. What about there was an event there in the Pacific. I don't know if you know this. It's called the blob, the warm blob in the Pacific. So it affected phytoplankton and zooplankton a lot in that area. I think in between 2015 and 2016. So there was a lot of acidification. Sea surface temperature was very high, and primary production decreased. But this was in between California and southern Canada. Do you think that could have affected the whales? It doesn't look like that, right?
No, I don't think so, because they only feed in the Arctic.
They don't feed on the whale.
No,
never.
no. So there are some instances that have been recorded where they feed, but overall they don't really feed along their migration. No.
Okay. I think that's all from my side. Thank you very much.
Thank you.
So thank you for the presentation and congratulations also for the nice work and how you presented and explained the work. So be proud of that. Okay, continue with some of the questions of my colleagues. Regarding this migration that you mentioned, because it comes a question right after one of your answers. So now with the climate change, so the migration is longer, and it's going to become longer. So we can expect that probably the estimations that you are doing now, you need to change. Probably this threshold that I show in one of the slides that you establish as a minimum point that in order to be successful in this process, so probably should change, because if they need to do longer distances without eating, so how it can affect the population? Yeah, I think that, so this threshold first needs to be tested too. So we would need to go to the Arctic and take measurements of individuals and kind of wait for another unusual mortality event to see if we can use that data in our threshold to predict that unusual mortality event. And do you have any data on the mortality associated with your just say indexes? I mean that in order to know really if you are expecting that in a given year, I don't know if you have an estimation, because I don't know how many individuals did you include in your analysis?
So for the threshold, it's around 10 to 15 maybe. It's a very limited data set, but overall, the grey whale population is, I think, around 20,000 individuals.
Okay. And you have any data just associating, I don't know, from your estimation of this given size, so I can more or less extrapolate of what are going to be in these 20,000, the numbers. and then, I don't know, with mortality records or whatever, just find an association that really this kind of stressors work?
Yeah, I think that would be the next step. Okay. So we didn't really include that in my thesis yet.
And then another point, quite important in this estimation, I don't know if there is, probably there are possibilities, I don't know, there is an additional, let's say, I am a ecotoxicologist, I come from another field, and this kind of changes in some, I come more from fish, but in the, let's say, body condition or let's say reproductive condition, at some point is associated also with accumulation of certain contaminants that can produce, for example, that those reproductive females, they accumulate more contaminants, and at the end, so maybe the descendants are not good enough, they are not fit enough in order to do that. So, do you think that this, I mean, that especially in lactation, because at the moment that the contaminants are translated from the mother to the calves in this case, or the offspring. So, any data, or have you checked on that, or any idea that can do some kind of this analysis in order to see if the reduction in certain, let's say, lactating females affect the health of the offspring? Yes,
I'm sorry. We also didn't really look into that either, but I did know that. So if females a decrease in body condition enough, they will first not stop supporting their calf because they want to survive themselves. So yeah, that would be my answer to that question.
Okay, thank you.
Hi, Julia. So let me just. So are there other whales that behave the same way when they have to do these long migrations?
Yeah.
Okay, which ones?
The humpback, the sai, the blue, they all go through fasting and feeding periods.
Okay, and so I suppose that affects their survival.
Yeah.
Okay, so what does it mean for us to know that you also found that out? What does it mean for conservation in general?
Well, it means that we need to protect their feeding areas and enable them to access these feeding areas and not exploit them. Or, I mean, there's a lot of research on noise, for example, and now that the sea ice is retreating, there's more commercial vessels going through the Arctic, so maybe that should be regulated in some sense, so that these animals, especially during the feeding period, are not interrupted.
Okay, but they are feeding.
Yeah.
I mean, it's the fact that they are able to, have their calves and everything. Mm-hmm. So the aspect of not feeding during long migrations and having these big, die-offs, mass die-offs. So that happens in a lot of species. Okay, so I just kind of wanted to know, what your work is adding to that.
Yeah, so the threshold is the main part of my work. So we can go to the Arctic and we can measure gray whales and we can predict these unusual mortality events.
Okay. How would you be able to predict them?
Well, so with our threshold. So we can go there, we can measure them, and if we measure enough individuals and we see that they're all kind of below this threshold, then we can say, or we can predict, that there will be an unusual mortality event happening within the next year or two.
Okay. Yeah, I just want to know in terms of conservation again, has there been any work to explain why they don't feed on migration?
I think it's their life cycle. I know that's how they go on their migration.
But has there been any work looking at that?
No, I just know that
you said sometimes it does happen that they would feed, so there are occurrences of that.
Yeah, so it's opportunistic feeding, and because also they primarily feed on benthic crustaceans, like I said, but now a couple of months ago there was a paper that was released where they observed gray whales feeding on fish actually near San Francisco. So all of this is pretty new and we're still kind of, looking more into it.
Okay, thanks very much.
Thank you.
So welcome, Jonathan. This is our last presenter of the whole symposium. So yeah, good luck and thanks for waiting all this time to present.
Yeah, thank you very much. Yeah, I have to say, when you stay with me, I have baby clownfish pictures in the end. So to start off, yes, I did my thesis with the University of Florida, and I was looking at dietary preferences of marine ornamental fish larvae during their early larvae phase, and I was using fluorescent microfields to track their feeding behavior. I guess I need this one. Okay, so to give you a bit of a background of the whole ornamental trade and the aquaculture, ornamental fish are fish that people want to have in their tanks because they're pretty, and we all know Finding Nemo. And there's actually quite high demand for those fish, especially in the States and in Central Europe. And unfortunately, this is also linked to negative aspects, which are mainly the high die-off during the transport of the wild captured fish, as well as the pressure on the wild stock, and then there are like some harmful fishing techniques involved, like you can see over here. Luckily, there's a way to bypass this whole problem, which is called aquaculture. And out of the 4,000 species that can be found in the trade, about 300 already have been successfully aquacultured. But you have to take this with a grain of salt, because successfully aquacultured doesn't mean you can find them in the trade. It only means they have been cultured in the lab mostly, and often it is not feasible to culture them, and so many are still wild captured. And my thesis is setting on the first feed. That's what I was dealing with. And the first feed actually is a bottleneck in the whole larvae culture. And to give you a bit of background, fish larvae are really small when they hatch. This is a clownfish larvae that has around 3 millimeters at the day of hatching, and they start feeding right away. So they need really tiny food, and it's really important to give them the right food, otherwise you have really high mortality. And the food that you usually give them is zooplankton, and because it's commonly found in coastal ecosystems. And if you select the zooplankton as first feed, you have to take into account the size, because the fish larvae needs to be capable of consuming it. Then, of course, there are some plankton types that have really high evasive behavior. This is also an important factor. Then it needs to be feasible to produce it in high amounts, otherwise the hatchery won't use it. And of course, the nutritional value of the plankton is also important. So, to start to talk about my thesis itself, I was looking at different plankton types and was feeding them to different fish to see which one they preferred. And the very first one I used in my experiment is the rotifer, which you can see down here. It's a considerably big plankton with 135 microns, and it has a moderate nutritional value, nothing too good. And what I still have to say about this is the industry standard, so it's used since decades, and you can find it as first feed in most hatcheries. And then you have the copypot malware, which are emerging because they are used more and more, and they have the advantage that they have they're a little bit smaller, so they can be consumed by smaller fish larvae, and they also have a really high nutritional value. And a disadvantage is they have a really high predator avoidance technique. So, and the last one I included into my experiments were ciliates, also highly abundant in coastal ecosystems, and they are even smaller than the copypot nalpier, have no predator evasive behavior, and the nutritional value is kind of uncertain because it's not used. So now you know the plankton types. Now to talk about the fish I work with. That's the first species, the yellow domino damsel. It's an ornamental fish, and the larvae is around 2 millimeters big. It's not available as a captive bred, and I was actually aiding a producer who has spawning broodstock that is struggling to culture the larvae, so they provided me with the eggs with data that I find out what they prefer to feed on. And I conducted an experiment at 1 day and at 3 day post-hatch to assess their dietary preference at this stage. Then I was looking at clownfish. The spine-cheek clownfish is also an ornamental fish. It's with 3 millimeter, a bit bigger than the yellow domino damsel, and I conducted the feeding experiment at 0-day post-hatch. And last one, the pigfish. Well, I'm not going to try to say this one as an ornamental fish. It's a bait fish. It's still my thesis, and talking about the size is something between the clownfish and the damselfish, and I conducted the experiment at 3 days post-hatch. And the whole idea of this was to assess their dietary preference at these stages. And so now that you have this background, I will talk a bit more about the methodology itself. And I was using micropheres, which are microplastic beads with a diameter of 1 micron, which are fluorescent. And I was incubating them together with the 3 plankton types, and they would ingest them, so I could mark them with the color, which would be later on visible in the gut of the fish larvae, which then selected whatever they preferred. And important to say is that you could only see them under the fluorescent microsphere, so for the fish it was just a plankton type, there was no color involved for them. And the experimental setup you can see over here, I had 20 replicate tanks, each has 40 liters, had 40 liters, and for each of the 4 treatments, which were 4 different combinations of prey type, I had 4 to 5 replicates. And the stocking density was varying between 2 and 50 larvae per liter, depending on how many larvae I had. And and at the end of the experiment I was sampling an average at least 20 larvae per tank, in some cases even up to 50. So I had like roughly 2,000 fish larvae analyzed. To give you now an idea of how this all looked like under the microscope, what you can see here is the gastrointestinal tract of the fish larvae. Here you have the yellow domino damsel at 1 day post-hatch, at 3 day post-hatch, the clownfish and the pigfish. And what you could see in the end was not the prey type itself. What you could see is only the micropheres and the color of the micropheres. And in this case, for example, you can see there's a blue spot, you have blue spot, the red and the green, which are all representative for the different prey types. And I have to say I was not trying to assess the quantity of ingested prey types, and was just going for absence or presence of each prey type for each larvae. Talking about the statistical analysis, I used logistic regression to compute odd ratios with confidence intervals of 95%, and then I did pairwise comparisons within the treatments between the prey types using Turkey HSD. And the different variables I included are over here. I had the binary output variable feeding response, which was either absence or presence of each prey type in the gut of the larvae. And I was, for the yellow domino damsel, including the treatment, the day of hatching, the prey type nested in treatment, and 2 interaction terms. And for the spine cheek and pigfish larvae, the model was simpler because it was only looking at 1 stage. So I only had the feeding response to the treatment and the prey type nested in treatment. So, talking about the results, the yellow domino damsel at 1 day post-hatch, what you can see over here are the 4 different treatments, which are the 4 different combinations of prey type. Here you had, for example, celiacs and naupere, celiacs and rotifers, naupere and rotifer, and all 3 combined. And the lowercase letters are obviously indicating statistical significance within the treatments. And then in this graph you can see the composition of the gut content, because the larvae often consume more than 1 prey type, or no prey type as well. And then here you can see the odd ratios, which are the output of the logistic regression. For those of you who are not familiar with odd ratios, for example, here you have the first treatment, celiacs and naupere, and the odd ratio is contrasting the possibility that 1 prey type is consumed over the other. Meaning if you have an odd ratio of 1, they equally prefer the 2. If it's higher, like in this case, they're 3 times more likely consumed a ciliate over naupere. And well, what you can say in the end about the results is that we have a really clear hierarchy and preference, starting with ciliates, which are the most preferred one, followed by the naupere, and the least preferred were the rotifers. And this is consistent throughout all the treatments. Staying with the same species, but moving on in the age, going to 3 day post-hatch, We have again here the feedings preference, then we have the composition of the gut content and the odd ratios. And what you can see here is that we have the nuclear and the ciliates preferred over rotifers, but we have to, we can see here that they here show a slight preference for the ciliates over the nuclear, while they here prefer the nuclear over the ciliates. However, the two are preferred over the rotifers. And the spine-cheek clownfish, again, the same way of presenting the data with the preference, the composition again, and the odd ratios. And we have basically the same finding like for the yellow domino donzel at 3 days post-hatch. We have here the clear preference for nuclear over ciliates. Here they're equally preferred, but overall we can say they prefer nuclear and ciliates over rotifers again. And the last species, the pigfish, we have again the preference within the treatments, the composition, and the odd ratios. And we have again a really clear hierarchy in preference. They prefer the nuclear followed by the ciliates, and again the least preferred are the rotifers. And so coming to the summary of all of this, it is really interesting to see that the rotifer, which is the industry standard, is actually the least preferred prey type. And this might be to the bigger size, so the fish maybe are just like having a harder time to consume this prey type, and so maybe size grading might help. But moving on to the copepod now here, the results are highlighting that this species has a high potential as a first feed item, since it was preferred by basically all the 3 species, at least at one of their stages. And the probably most interesting finding about all of this is that the underdog, the ciliate, is actually preferred by the spine-cheek clownfish and the domino donzel at first feed, which highlights their potential as a first feed and maybe make it even possible to feed really small larvae that are not able to feed on the copepod nuclear or the rotifers. And so the overall conclusion of this is that we have to be aware there are restrictions of preference, which are physically, just simply they're not able to consume them because their mouth gape size is too small, or they have a hard time catching them. And this, of course, also a question that we have to ask is that, is preference necessarily indicating the best suitable one, or do they maybe don't know what's best for them, in other words? And as I already mentioned, the smaller stage prefer the smaller prey type, which is interesting. And of course, since the ciliate was preferred, the next question we have to ask is, what is the actual nutritional value of a ciliate? Or can we maybe even increase the nutritional value by maybe enriching them like we did for rotifers? And that's all of it. And here are the promised clownfish pictures, and I want to just and I just want to acknowledge all the people that supported me during the thesis, and also the companies that provided me with the eggs, and thank you for your attention.
Thanks very much, Jonathan. Does anyone have any questions in our audience? Loads of questions. Okay, the start here at the front, yeah?
I was wondering, so we didn't do anything on the mortality rate associated with each?
No mortality.
Yes?
I was just wondering, how do they feed if they haven't even hatched yet?
Oh, they hatched.
Oh, they
one day
okay,
post hatch means they hatched 1 day ago.
Oh, okay, so 0 is the day they hatch.
0, yeah, that's actually interesting. That's good to know, because 0 day post hatch is from the moment of hatching until 24 hours, and then 1 day post hatching is from 24 hours to 48, and so on. So it is a bit confusing. Yes, I know.
Thanks.
Okay.
Yes.
Yeah, I was wondering if a problem with the copepods is their evasiveness. Can you remove that or immobilize them somehow?
Actually, there are studies on that, or one study on that. I think they tried to pose them to different salinities and to see if they could decrease the evasive behavior. But I think it didn't really work, and I'm also not aware of any application in an industry of that.
Do you have a question as well? No? Yeah.
How long do you let them feed before looking at the big compare?
So for the domino damsel at both stages and the clownfish it was 4 hours, and for the pigfish it was 4 hours. I also have it. Okay, wait. I have it here, yeah. So for there you go, yes. Pigfish 2.5 hours, and for the others 4 hours. Yes. I was first going for 2.5 hours, and then I saw that this was more than enough, and then I reduced.
Anybody else? Okay, so our panel.
Okay, so congratulations for the work and the presentation. And so thank you for the way that you introduce this topic, and you introduce your results and you present, and I think that make coherence with the later conclusions that you take. So congratulations for that. I have one question, maybe it's because you are the last one after these 2 days. No, it's one of the graphs that you show, the stacked bar that you have in all the...
Oh yeah, wait, let me find it. I don't know if you can switch to
this one. Okay,
yes.
You say proportion of ingested prey type per combination. So,
per combination.
So what does mean? So how you calculate that, or what is the meaning of that?
Yes.
Because you have the proportion of ingested prey types,
yes.
Percentages.
Yes.
And then
yeah, let me explain it. So as you can see here, you have 100%, and here you didn't have 100% because not all of them were feeding. Actually, a quite big portion was not feeding, and you can see that roughly 10% had only ciliates. Then like a really slim amount had ciliates and outlier, and the greenest and outlier, which were like 5%. And if you go here, you can actually see that the ones that had both are basically like the bottom one, and then you have the ones that only had ciliates, and then the one that only had an outlier. And I think it was just, I wasn't including it into my statistical analysis, but I think it was just interesting to see and to display that there were different combinations. Okay. So yeah, makes sense?
Yeah, it makes sense. Okay, good. The species that you selected based on any special characteristics that differentiate each one in order just to test the different food, or something that they have in common, or was just they were available and you test them?
It was a bit of all of that. I went there to work on ornamental fish that then I didn't have eggs for a long time that I could use. So I ended up using the pigfish for my first experiment, and then I had the opportunity to get those eggs from the company, which was purely opportunistic in the end. But luckily they're both in the same family, so they're kind of comparable. The clownfish and the damselfish, yes. But yeah, the idea was to like focus on ornamental fish, because that's what the thesis is about. Yes. Yeah, and I would have liked to conduct the experiment with way more species, and also with more pelagic spawners, because the clownfish and the damselfish are demersal spawners, which are problematic in aquaculture, but not as problematic as pelagic spawners like the pigfish.
Mm-hmm.
So I would have loved to also include more of those. And actually, if we go back to the slides down here, there you go. This fish, the Harlequin baslavi, we managed to culture it for the very first time in captivity while I was there, and it is a pelagic spawner. And I was trying to do the experiment with this species as well, just because it was available, but the survival was just so bad that they wouldn't even come to first feed. Yeah.
Okay. I am very surprised that after 1 day per hatch, they are already able to feed.
To feed, yes.
Yeah.
Yeah, it is.
Several of in most fish, so the larvae is just during the first days, so they're really in the jaw sac. They don't have the mouth.
Exactly.
No intestine develops. So.
Exactly, yeah, that's the difference between demersal and pelagic spawners. Demersal spawners, they lay egg clutches, like patches on hard substrate, and they usually develop they hatch way more developed. And the clownfish especially, they hatch like little fish, like they are ready to feed the moment they hatch. And yeah, pelagic spawner like the pigfish, you may have noticed I only started feeding them at 3 days post-hatch, because that's the point when they start feeding. And that's the difference between them, yes. Yes. Yeah.
No more questions for myself. Thank you.
Well, Simon, congratulations. Very, very nice presentation. I really enjoy how you frame it. The diagrams, the explanations of the experiment are very nicely done. So congratulations for that. I'm surprised that you didn't use Artemia.
Well, Artemia is for first feed just way too big.
It's too big.
It's way too
Even
big.
the small Nauplia.
Even the Nauplia are way too big, yes.
Okay.
You can start introducing them for clownfish around like 5 days or even 7 days. Same for pigfish. So like they're already through this really crucial phase where they're too small to feed on Artemia and rotifers.
Okay, I see. And I have a question regarding concentration here, because
yes,
you are not interested in the concentration of the particles inside the animals. That's not the point.
That's a really good question.
That's not the point. But what about the concentration of particles outside?
That's a really good question. Yes, yes, yes. Yep.
If you go back to the graphs.
Yeah. You probably mean this one, right?
No, I mean the graphs.
The graphs. Okay.
I'm sorry.
This,
yeah,
those,
this one.
Yeah.
So in the treatment that you put the 3 ones, the ciliates, naupli, and rotifers.
Yes.
What is the concentration of these 3 compared to the concentrations of?
Absolutely, the concentration is higher because you have 3 of them. Absolutely.
You should have a control probably to account for the change in concentration.
I know, yes. That's a good point indeed.
Still, I think you can make something with that, of course. But, yeah.
Yes. And it might also explain why we have, for example, in this treatment, just like little differences, right? So this is actually the way I explain those variations.
Okay.
Yes.
Very well done and congratulations.
Thank you.
Thank you, Jonathan. Just one more question and we finish this. So you explained in the beginning that even though there's the potential for aquaculture of a lot of ornamental fish species, it doesn't really happen in real life. Would you be able to elaborate a little bit more as what are the impediments for this to happen?
Absolutely. I mean, the problem is that there are some species that are already entirely replaced the wild capture of the species. A good example for this are clownfish or dollybacks or like very few other species. But the whole problem that is underlying is that it is just more efficient to capture them in the wild. And as long as you can import centrifugal pottery for 50 cents from Indonesia, the aquaculture industry will never be able to compete with that. But of course, that's where the whole policy making gets important. We see things like in Hawaii where we have like restrictions on or bans on fishing, and that of course increases the value of the fish because people still want this fish, but it can't be wild captured anymore. So people are suddenly like willing to pay more for aquaculture fish, and of course, as long nothing is happening in this area, many species can never be cultured in a way that they can compete with wild capture. Yes.
Okay, so your work brings a bit of importance to perhaps the advent of that?
Yeah, I hope so, and I also hope so that, I mean, it is really important to understand how to culture those fish, with the idea to replace wild capture, and especially talking about we only have those 10%, even less 10% already successfully cultured, and the reason for that is that we don't really understand what they need in the wild, not only talking about the right plankton type they need. I mean, there are so many different plankton types in the sea, we basically just feeding them 2 of them or 3 maybe, which of course is not ideal. And yeah, I think by already especially highlighting the importance of ciliates and other smaller plankton types, this hopefully contributes to more breeding success in this field. Yeah.
Okay, thanks very much.
Thank you.
And this is the end. Congratulations on, finishing your masters for those that finished completely, and good luck to those that still have to submit in August. And yeah, just go enjoy yourself.